AI models to predict which molecules are likely to bind a target or have desired biological effect. Biology is a system of complex interacting networks. AI excels in modeling these complex biological processes, such as mapping intricate signaling pathways or understanding the dynamic of cell metabolic networks. This system-level understanding is critical for grasping how disease develop and how they might be treated. Let's look at a concrete example that ties together AI, drug discovery, and botanical therapeutics. In 2023, researchers sought to discover new senolytic drug compounds that can eliminate senescent cells. Traditionally, drug discovery screening could take years. So instead, the team trained machine learning model. They computationally screened various chemical libraries and validated the senolytic actions of gintilin, periploxin, and aleandrin in human cell lines under various modalities and of senescence. The compounds have potency comparable to known senolytics. This approach led to several hundred-fold reduction in drug screening costs, demonstrating that artificial intelligence can maximize the advantage of small and heterogeneous drug screening data, paving the way to new open science approaches to early-stage drug discovery. This is a striking case of AI bringing traditional botanical medicine and modern drug discovery together. It also illustrates a broader point. AI can enhance research on natural products. Similar approaches are being used to predict which molecules in, say, traditional herbal remedies are the active ones, or to suggest combinations of plant compounds that might work synergetically. In fact, AI tools can even predict synergetic effect of herbal components, helping to validate or refine multiple herb therapies. Overall, this case study highlights AI ability to unlock a value of botanical diversity in the search for a new drug. By finding promising molecules faster and more effectively than classical pharmacological screens. This approach has been widely adopted, resulting in proliferation of publications. Another exciting advance is AI-driven de novo drug design. Generative models, like variations of neural network, can invent new molecular structures with specific properties, essentially suggesting novel drug candidates that don't exist yet. These models learn the language of chemistry from databases of known compounds and can then imagine new molecules that might bind a target or have desired biological activity. Medical chemists can synthesize the most promising of these AI-proposed molecules and test them in the lab, accelerating the design cycle. In early 2020, the first ever AI-designed drug candidate entered the phase 1 clinical trial. This molecule was developed by Accenture and in partnership with a pharmaceutical company, and it processed from the concept to trial in just under 12 months. Since that milestone, several other AI-designed compounds have entered trials. By 2022, at least 15 AI-discovered drugs candidates were in clinical development across various companies. Areas range from oncology to fibrosis to rare diseases. This shows that AI is shortening the drug discovery time, helping to find and optimize lead compounds more quickly. It's even being used for drug repurposing by predicting different targets a drug might hit. Overall, AI is becoming a crucial R&D partner in pharma. It can handle the heavy lifting of analyzing data and generating hypotheses, allowing human scientists to focus experiments on the most promising leads. AI contributes across the full drug discovery pipeline, from early target identification to safety and toxicity assessment. In target identification, AI integrates multi-omics data to uncover disease-gene relationships, predict protein structures, and validate target using real-world and biological network data. Deep learning and NLP accelerate hypothesis generation beyond traditional methods. During lead design, AI enables virtual screening of massive chemical libraries, de novo molecule design, and optimization of structure-activity relationships. Generative models and reinforcement leading dramatically shortened hit-to-lead timeline. For admit prediction, AI models forecast absorption, metabolism, toxicity, and drug-drug interaction earlier, reducing late-stage failures. Hybrid approach that combines machine learning with physiological modeling improves biological realism and transitional relevance. Finally, in polypharmacology and toxicity, AI supports multi-target predictions, off-target risk assessment, and safety profiling, which is critical for complex disease and repurposed drugs. Network-based and ensemble methods help balance efficacy with safety. Overall, AI is not replacing experimental science, but augmenting it, improving efficacy, de-risking development, and enabling more rational data-driven drug discovery. While AI brings great promises, we must navigate several challenges and ethical issues to use it responsibly in biology and medicine. Bias is a top concept. Another issue is explainability. There is a big push in AI called XAI, explainable AI, to address this, but remains challenging. Privacy is also critical. Finally, there are accountability and regulatory considerations. It means we need rigorous validation of AI systems and clear guidance. Bodies like FDA are beginning to issue guidance for AI tools. The key is that human experts must stay in the loop. AI should assist, not fully automate, critical decisions until we are very confident in its reliability. By proactively addressing bias, explainability, privacy, and accountability, we can ensure that AI's integration into biosciences proceeds ethically and benefits everyone. The convergence of AI and automation is reshaping drug development timeline and outcomes. Self-supervised learning allows models to extract knowledge from vast, partially labeled biomedical datasets. Real-world evidence integration brings clinical records, genomics, and patient data into the discovery development loop. Adaptive clinical trials further close the gap by dynamically adjusting protocols based on interior AI-driven analysis. The convergence of AI and automations, where robotics, autonomous laboratories, and intelligent decision systems work together. This creates seamless end-to-end pipelines that dramatically accelerate experimentation, analysis, and iteration. The impact becomes clear. Traditional drug development timeline of 10 to 15 years are projected to shrink to 5-7 years, effectively halving the development time. Beyond speed, this converge enables personalized therapy, where the treatments are tailored to individual patient biology rather than the population average. Overall, this converge signals a transformational shift, moving from slow, linear development to adaptive, data-driven, and precision-focused drug development ecosystems. The future is interdisciplinary. As we move forward, a balanced embrace of AI, welcoming its power, while rigorously validating its outputs and binding ethical guardrails, will likely yield some of the most profound advances in biology, science, and medicine in the coming years. AI does not replace biology, it accelerates it. The real divide will be not between AI and humans, but rather between scientists who can work with AI and those who cannot. Thank you. And of course, now I'm absolutely delighted to answer any questions if you had time.
Thank you very much, Professor Dushenkov, for a very interesting and insightful talk. But before we proceed the question and answer session, would you please wait for a moment for the other speakers to deliver their talk, and then we can proceed the Q&A. Thank you. Now we move on to the Okay. All right. I think we can start to enter the Q&A session before we proceed the other talk. Please, any participants that may deliver the question before we proceed the other talk. Please, first 3 questions. Is there any question from the floor? Maybe before we have any question from the floor, I have a short question regarding the AI role. I hope you don't mind. It seems like AI has so many potential roles in biomedicine. Do you think it's going to be replacing the what do you call it? The user, or it's going to be helping the user?
Well, whatever I talked today about was just a general overview. And the development of AI tools is so fast and so powerful that basically anything that will go today for development may be already outdated tomorrow. It's a tool. It's a wonderful tool, and it definitely will help to speed up the discovery and development of pharmaceutical products. It just started to use for the natural products. However, the field is wide open, and I certainly hope that AI will be used for development of wonderful products and analyzing already products that are used by natural communities, for example, native communities. Also, we need to look for different approaches to AI. Not necessarily just using it as a calculus tool, just to calculate things, but give it a little bit of more freedom to produce initiative and suggest development in different areas. That's the area which is underutilized right now and maybe more useful than just calculation. Of course, as I mentioned, it's enormous power of analyzing the data. The scientists were struggling to identify potential of folding of proteins for almost half a century, and never went that far. Then AI was able to do it in a few years, and now we have the models which allows by sequencing of the amino acids predict the property of proteins. they're designing proteins and so on. AI could be used in a variety of things, which will never completely substitute the human. We need to apply human intelligence, we need to apply our ethics, we need to be careful because as I mentioned, yes, the AI has biases. The AI training is very powerful, but the question is what AI is training on. And there is a big rush to figure out that current AI will not be trained on AI materials. On the internet, for example, there's a lot of materials which are generated by AI. The conferences are filled with the, papers produced by AI. And it might become the self cycle. The AI will be trained on AI. And we may lose a lot of opportunity there. So, of course, there are ways to prevent it, and the experts are working on that. But to answer your question directly, AI is a tool, very powerful tool, and we have to use it carefully.
Thank you very much for your answer. It's very, very insightful. Thank you. Is there any question from the floor, please? Okay. Nggak ada Bapak Ibu? Pertanyaan, silakan. Okay, if there is no question, I guess if you want to stay here, it's fine. But if you want to do something else, please don't mind. Thank you. Thank you for your time.
Thank you very much. Thank you very much for attention.
Thank you. All right. Now we move on to the second invited speaker. I think I want to address, before we start, Dr. David Bedokian. Are you here? Belum. Cek email. Email ada nggak? Huh? Oh, okay. Maybe we can next move on to the second invited speaker with Professor Siti Efliza Ashari. Are you with us right now?
Yes, I'm here.
Okay, thank you. Let me share Professor Ashari's brief biography. She is an associate professor at University Putra Malaysia, UPM, specializing in industrial biotechnology, pharmaceutical sciences, and statistical experimental design. She earned her PhD in industrial biotechnology from UPM with research training at Complutense Universidad de Madrid, Spain, and sabbatical experience at Durham University and Hawke York Medical School, United Kingdom. Her research spans chemistry, biotechnology, nanotechnology, and product innovation with a focus on drug delivery systems for wound treatment and cosmeceutical development. Her notable innovations, including BioEase Oleate and a kratom patch, have earned multiple gold medals and innovation awards, reflecting her commitment to translating advanced statistical modeling into impactful therapeutic and commercial application. Please give a warm welcome to Professor Ashari. Time is yours.
All right. Okay. Can you see my slide?
Yes. Yes, we can.
All right. So, all right. One moment. Okay. So, is it in the slide mode? Is it in a full screen now?
Yes, yes.
All right, okay. All right, okay, thank you very much. First of all, to the moderator, Dr. Vivid. So, I'm Siti Afliza Ash'ari from Faculty of Science, University Putra, Malaysia. So, I will be presenting part of my recent work, which is on a novel approach for wound treatment using Mitragyna spiosa, encapsulated nanostructured lipid carriers in hydrogel film. All right. So I think I am the only one participant from Malaysia. So I think I will prefer to introduce a part of our university background. So based on QS World University Ranking, so we was ranked in second positions in Malaysia, and in the fourth place in Southeast Asia, and in Asia we are in rank 20th. So our expertise, our pioneer is basically on agriculture. University Putra Malaysia, we are actually in the first pioneer for agriculture and forestry, which is our first in Malaysia, and we are in 75th in the world. Alright, so the rest will be our expertise is based on veterinary science, petroleum engineering, and also for economics and econometrics. Alright, so since this is particularly on natural products, so I think that is the best platform for me to share part of my research work recently. Alright, so I will begin my talk with our motivations and the challenges of the kratom, and we are using drug delivery system, and I will share part of our finding based on that work, and I will finally conclude the rest of the presentations and share with you some of our achievements so far. Alright, so basically, one of our motivation is because of the Malaysia. Malaysia healthcare, so in Malaysia healthcare budget in 2025, for the year in 2025 is never static. The budget is always increasing. So Malaysia itself, for example, our Ministry of Health Malaysia, which is the second largest among other ministries, allocate around 45.27 billions purposely on the productions of healthcare. So in 2030, Malaysia with new industrial master plan or NIMP in 2030, we will promote integration of value chains for better local productions of active pharmaceutical ingredients or API. So with that motivation, we are looking forward on the one of the active API, which I think that will be this plant is actually very famous in Thailand and in Malaysia. So the name of this plant is Mitragyna spiosa. Its origin in Southeast Asia. The family name is Rubiaceae. Local name in Malaysia we call it as ketum. In Thailand, they always use the words as kratom. And I think in Indonesia, they call it as biak-biak. I think that one will be familiar with all of you guys for this plant. Okay, so this kratom or this ketum, in traditional uses, it can be used as diarrhea, muscle pain, can be reduced coughing, diabetic, and also for wound healing. Activities, it can be used as antimicrobial, antioxidant, antimutagenic, and of course anti-inflammatory and anti-nociceptive. Alright, so this is the part of the active ingredients that we are looking forward as our API. Alright, so what are the challenges of this kratom? Okay, actually in traditional uses, this kratom is used as a poultice. Back in 90s, they used this plant as a poultice in order to treat any rashes, any inflammatory work, or any for wound healing. But then, because of these properties, this plant, naturally, it has a low biocompatibility and large droplet size of MS, which is making it difficult to penetrate into the skin. And in terms of storage, it is unstable during long-term storage for wound healing application, and also it actually have a low control administration for transdermal drug delivery. So it comes to our work where we are trying to do some innovation where we transform this active API alone with these patches in a patches product. Yeah, so we are transformed into a drug delivery system. So what is drug delivery system? So basically, drug delivery systems are developed to effectively deliver the required amount of drugs. So now we are talking about our API drugs to appropriate target site and maintain the desired drug levels. So we have a lot of types drug delivery system. Okay, for example, we have nano emulsions. This is very common where we, from our previous experience, we develop a few products using nano emulsions, and then we have liposome, niosome, ethosome, solid lipid nanoparticles, NLC, polymeric nanoparticles, and nanofibers. So in our work, we are looking forward on NLC, nanostructured lipid carriers. Alright, so how do this work? This is the mechanism where we have if you can see this mechanism, we have the drug, which is our API, our MS, kratom, crude extract kratom, embed with NLC system. Okay, so for example, here we have solid lipid, the one with green color, and then we have a liquid lipid, and also we have the surfactant as emulsifier. Okay, so basically a nanostructured lipid carrier produced using emulsifier, solid lipid which is biodegradable, and the size range is between 50 to 1000 nanometer that we are looking forwards on the particle size so that it can be penetrate easily in our skin. So another advantages by using NLC is because of its high drug loading and drug release, its low toxicity, and then it's having a small particle size which are able to penetrate into deep skin cell easily, and of course this is mimic our natural living tissues in the skin. And also we try to convert it into hydrogel film for wound dressing purposely, because in hydrogel film it can provide moisture content, it can improve the wound microenvironment, combat infections, facilitate the healing process, and also it can give a cooling effect. Rather than we just use the kratom itself in plant form, it's better for us to transform it into a hydrogel thin film. So that would give a patient comfort. Alright, so for wound healing, very prominent characteristics in order to produce a good product, in order to treat wound, the ingredients in the formulation should have antioxidant, antimicrobial, anti-angiogenesis, and anti-inflammatory. That is the prominent characteristics for wound healing. Alright, so we have a very extensive method in order to do some formulation, where we start with the preparation and optimization. Okay, so we start with the extraction of our plant, extract the crude extract, and then we do some screening, and then we do some optimization. So at this part, we managed to publish. We secure one trade secret where we have to secure the formulations before we move on to the other publications. And then we move on to the behavior examination. We do some preparation. We check on its gel fractions. We check on its rheological behavior. And we following with the physical-chemical characterizations, because we are chemists, so we want to identify the structure of our formulations, which involve the phenolic alkaloid. So we want to check the presence of the active ingredient that can treat the wound healing. Alright, then this is the most crucial part where we have to do some evaluation on wound healing. For example, we have to check on its antioxidant, antimicrobial, anti-inflammatory, in vitro cytotoxicity test, and in vivo and in vitro analysis. All right. So we come up with the first, with our first paper, our first part. First thing first, we need to do some standardized crude extract. So we successfully published one high impact factors of this journal, where we managed to identify about 27 compounds using LC-MS, which we think that all these compound, we have the spectrum, but then we can just look forwards on this paper later on. So all these 27 compound having characteristics on the promoting the wound healing, such as alkaloids, flavonoids, phenolic, and fatty acids. The one that is very crucial one is actually in Metrogyna spiosa. We have a very abundant active compound, which we call it as mitrogynin. Okay, as you can see here, based on the literature, mitrogynin itself contain about 60%, and the other will be other compound. So, but we are looking forwards on this mitrogynin and also the other 26 compounds which related with the wound healing. So we managed to convince that these plant materials having the API that having a characteristics of wound healing treatment. Alright, so next we move on to the formulation. So after we extract the crude, the crude plant, so we do some formulation where we transform the crude extract, the one in the solid solution, the one with the solution, with the like a cream or semi-solid formulations, okay, encapsulated in nanostructured lipid NLC system. So in this work, we also, one of my PhD students managed to publish in another one paper, which is high impact factor in industrial crops products, where at this stage, we successfully produce a very stable formulation. Okay, and in the first place, we have to do some screening on the liquid lipids. So we have a lot of lipid testing. We have almond oil, sunflower seed oil, ricinoleic acid, soybean oil, oleic acid, and virgin coconut oil. So based on this, we are looking at in terms of its solubility. So our crude extract is solubilized better in oleic acid. It's about 2.859 milligram per ml. And in terms of its solid lipid, we are also testing with several solid, such as we have compritol, stearic acid, trilaurin, and trimyrostin. And copritol or COM give a better drug entrapment for the formulation. And also for the surfactants, we have twin 20, twin 40, twin 60, and twin 80. And twin 80 give a good highest transmittance. Selections of HLB and percentage of transmittant about 51.53. So by looking at this microscopic evaluation, we can see this is the first preliminary observation where we can see that the whole formulation, our formulation, is what we call is in a good conditions, where we can see there is no agglomeration, so it's very unstable conditions, and also in terms of its particle size as well. Alright, so you can go further on this paper because I just picked I just highlighted the most important part on this paper, on this work. Okay, then we proceed with the fabrication, the characteristics, physicochemical characteristics. So we have to check the particle size, and it's confirmed by we compare these two samples, MS-NLC and MS-NLC in hydrogel. We can see that both of these samples still in nano size, which is suitable for transdermal delivery. And also for zeta potential, it's in a stable range between 30 to 33. And for PDI, PDI shows less than 1, meaning that our formulation, our particle size is a uniform distribution, and the particle dispersion is homogeneity. Alright. So then we have looking at the surface morphology based on these two. It shows that the shapes using transmission electron microscopy is in spherical shape, which is in a smooth surface. In terms of storage stability, our formulation, our end formulation is stable in under stable condition with different temperature within 90 days. you can see that we maintain the particle size in different temperature, minus 20 degrees Celsius, 25 degrees Celsius, and 45 degrees Celsius. Both of all of this, we maintain the nano size, which can explain by the migration of the solid lipids and also for the liquid lipids toward the system. All right. So we can see here with different temperature, the particle size still in our considered region in nano size. All right. So then we move on to the very simple analysis. What can I say before we proceed with the most critical work on wound healing? So this is a very simple preliminary work on scratch assay in vitro, where we use 3 T3 fibroblast cells. So we compare with 4 samples, one with the control, one with the MS extract alone, and then one with MS NLC, and also with MS NLC in hydrogel film. So we check through the wound closure. Alright, so for 0 hour, there is no closure at the moment, and then after 24 hours, okay, we can see here there is a closure about, you can see here, this is MS extract. Alright, so this is the MS NLC percentage of wound closure. So you will see, for MS NLC hydrogel, okay, this graph, after 24 hours, we can say about 64.21% closure of the wound by using fibroblast cell 3T3. So we can say that actually it can give actually excellent improvement, and they will give a strong proliferative and migration activities towards our formulations. Alright, that can be increased to cover the scratch wound area. So you will see this is the scratch wound area. So after 24 hours, about 64.21% improvement on the wound closure. Alright, so that is in terms of scratch assay. And then we move on to analyze the nanotoxicity test using zebrafish. So from here you can see in terms of its toxicity profile. So zebrafish embryos exposed to high concentrations, about 500 mg per ml. All sample tested. So we have 1, 2, 3, 4, 5, 6. So this is the zebrafish embryos. You will see there is a mortality rate here, the heartbeat for each of these samples. So we can see it's in normal rate after we apply the formulation. Okay, it's about 156.75 heartbeat. The hatching rate, the normal hatching rate is about 48 to 72 HPF, and also there is a movement of tail coiling increasing. Okay, so we can see based on these results, based on this profile, all the sample tested, there is no significant signs of development abnormalities such as bend or undetached tail or any spinal column curving. So it's normal when we apply our formulation. Alright, so that is in terms of toxicity test using zebrafish. And then we move on. So this is our latest finding, where we can see there is improvement when we use sprout dolly rat. See, we check on its wound closure as well. So in this particular work, one master student managed to publish, not publish yet. They use this sprout dolly rats. So we will see after, this is from 0 days to 21 days, we can say that after 21 days, in different concentration of MS NLC hydrogel, the wound also decrease, completely close the wound here, the wound areas. You will see there is a start formation of, you will see this is the morphology. It's very, look at the diagram here. So we can see at day 21, you just refer with this picture. In this day 21, you can see that the rat skin, there is a start formation of collagen. Okay, you will see there is a collagen here. So HR means it start to have a hair follicle, the growing of hair follicle, in order to close the wound. Then we have, this is the stratum corneum, this is the skin. And also we have the mature collagen. As you can see here, there is the collagen perform, completely collagen formation and wound closure were produced in day 21. Okay, by using this different concentration of MS-NLC hydrogel. So that is our recent finding on the productions of Mitragyna cipiosa, for embed in drug delivery system. So as a conclusion, I would say that our kratom or Mitragyna cipiosa encapsulate NLC in hydrofilm possesses beneficial effects which play a crucial role in wound healing. In terms of its physicochemical properties, it shows that excellent stability with maintained particle size with less than 200 nanometer. And because this is a very new, novel approach, so I think that particular finding can help us to give, to have a more evidence that can promote wound healing with improve its pharmacological and toxicology properties. So this is the first phase of our recent, because we have, I think we have a lot of analysis later on, but then I'm not showing in this talk. So this project is actually started in 2018. Alright, so this is part of our research achievement, where in 2022 we successfully produced one IP, which is a trade secret, on the new formulation of Mitragyna cipiosa. And then we secure one grant from Malaysia, from Ministry of Higher Education, MOHE. And then we received a gold medal in 2023, and we won top 10% achievement in third Cannes scientific competition in Iran in 2023. And recently, last 2 months, we won gold medal in invention and innovation design exhibition IDEX in Malaysia. All right. So this is our firstborn, what can I say, in 2024. This is my student from 2018 until 2024 last year. So we managed to publish around 8 papers specifically on 2 projects in terms of using of Mitragyna cipiosa. The one with my first student, Dr. Fazila, she did on Q-bosom. Another one, Dr. Sharifah, she working on NLC. All right. So with that, I would like to thank our students. We have three PhD students, and then we have the collaborators in Malaysia, and also international from Japan. My collaborators in Japan, she helped us on angiogenesis work, and my colleagues in UK in Durham University, he's working on lipid synthesis, and also the one that helped me a lot on wound, because I'm a chemist, because wound is actually something new for me. So one of my collaborator from Hall York Medical Center, Dr. Holly Wilkinson, she helped me a lot on the wound, in terms of its very general informations, and also hands-on using fresh skin. So I have the opportunities to work with them in order to strengthen our end product to be, I think we cannot commercialize that because we just finished on the first phase. So perhaps we are looking forward on the next collaborator, which can give us funding to proceed with the human skin. All right. So with that, that's it. Thank you very much. I will open for any questions.
Thank you, Professor Asari. What a remarkable research and very significant potential to promote wound healing from MS-NLC hydrogel film formulation. Congratulations. Very
Thank you.
tremendous work. So before we proceed the Q&A, please hold for any questions. We want to move on for the third speakers. For the third speaker, we will invite Dr. David Bedokian. Is it ready for the video? Let me share his brief biography. So Dr. David Bedokian is Vice President of Sales at Bedokian Research, a global manufacturer of high-impact flavor and fragrance ingredients founded by his grandfather, Dr. Paul Bedokian. With over 2 decades of commercial and strategic experience in the flavor and fragrance industry, Dr. David leads the company's global commercial strategy, distributor partnerships, and customer development initiatives across the Americas, Europe, and Asia Pacific. He oversees customer engagement and collaborations that connect formulators with Bedokian's expanding portfolio of nature identical and sustainable aroma and flavor ingredients. Working closely with R&D, production, and regulatory teams, Dr. David ensures precision and reliability in bringing new products to market while maintaining the company reputations for quality and innovation. His active involvement in industry organizations and scientific consortium reflects his commitment to advancing collaboration and innovation within the global flavor and fragrance community. Without further ado, please welcome Dr. Bedokian. For your time is yours.
Our business was founded by my grandfather, Paul, and he worked in the industry for years before founding the company, and his big claim to fame. Our business was founded by my... Hello everyone, Selamat pagi. I'm David Bedokian. Very sorry I can't be there in person today, but happy to be joining remotely to introduce our company. We're very excited to be a part of this consortium and work with everybody here. And just for fun, I'd like to add, my wife is actually Indonesian, so I typically go to Jakarta and Bali once a year. So hope to... Our business was founded by my grandfather, Paul, and he worked in the industry for years before founding the company, and his big claim to fame was actually the first person to synthesize and commercially sell the smell of grass, which is cis-3-hexanol. And our big focus was always on specialty ingredients as well, very high-impact materials. Our first commercial success was actually Charlie Fragrance. My grandfather had identified and synthesized 2 ingredients back when regulations were much easier. They smelled great, gave them to his perfume buddy, who threw it into his mixture, and this iconic fragrance was made, and that really got our company off the ground in 1973. Today we offer 250 high-impact F&F specialty ingredients, including a growing number of sustainable ingredients, which I'll talk about more later. And we really try to focus on products that are generally non-commodities, that are unique, innovative, pure, and high impact. And we have a team that does presentations and takes these around globally to all the multinationals and flavor fragrance houses around the world. Our company is headquartered in Danbury, Connecticut. We also have sales offices in Switzerland and Singapore. So I expect our Singapore representative will be joining next time. And we have over 170 employees, a very long tenure. Our longest employee had served with us for over 49 years, which we're very proud of. And we really like to say we're a technology company as well. We don't just do science, but we really find innovative solutions to get the right products to the markets. While we haven't currently focused too much on the natural space, we are really shifting gears as a company and really focusing on sustainability. So one of our big initiatives is launching many products that are synthetically made, but from renewable carbon starting materials. So there is a big shift in the markets, and us getting more toward that is very important to us. And another reason we'd like to join this and be involved in this consortium. One thing that we believe also sets our company apart from others is our distillation capabilities. We have very fine distillation. We can separate very challenging fractions to get specific molecules, deal with unstable mixtures, and especially in the natural side, we do a lot of toll work where we're getting the odor right, making blends, and doing things that are very difficult to do. So that's something that we love to do and is our specialty. And I think that'll be very important to contribute to this group. So to bring this all together, I think our main interest in this consortium is to help the teams, work with the teams, try to identify unique oils that might have high value, add our analytical and regulatory experience, and also our fine toll distillation capabilities to really support exploring how to commercialize these products and to take them to market. We're also eager to collaborate with our members to see are there any other ways we can support them or find partnerships or add any scientific value to this. So we're very excited to be a part of this and thankful to have been invited. So thank you to Ilya and this team for having us be a part of this, and looking forward to see you all in person and have our teams interact with everybody in the near future. So to conclude, feel free to check out our website, Badukian.com. We have a lot of interesting things there, including our products, samples, an iCatalog to find our ingredients, and most importantly, my contact information, david@badukian.com. So again, we're looking forward to working with the scientists to identify and help work with many different oils, for collaborators to help with any toll partnerships, and any way we can support this project further. So makasih, thank you very much. Have a great rest of the consortium, and hope to see you next time. Take care.
All right, thank you, Dr. David Badokian. We move on to the last speaker, Professor Muhammad Asif Khan. He is an Associate Dean at the College of Computing and IT, University of Doha for Science and Technology in Qatar. He earned his PhD from the National University of Singapore before joining Johns Hopkins University, USA, as a faculty member. He later became part of he became part of a pioneering GHU, or Johns Hopkins University team, to establish a collaboration with Perdana University in Malaysia, where he served as the founding dean of the School of Data Sciences and founding director of the Center for Bioinformatics. Professor Khan was also a visiting professor at Besmilem Fakih University, Istanbul, through Turkey's International Fellowship for Outstanding Researchers. He is currently the Vice President of the Asia Pacific Bioinformatics Networks, or API-BioNet, and the chairperson of the Executive Board of the Global Organization for Bioinformatics Learning, Education and Training, or GOBLET. His research focuses on biological data warehousing and bioinformatics applications in virus study, immune responses, vaccines, drug design, and disease biomarkers. It is my pleasure to welcome Professor Khan. The floor is yours.
Good morning, everyone. Hello from Doha, Qatar, and I'm sorry if my voice is a little soft because it's very early in the morning right now, around 5, and some family members are sleeping, so I'm going to be a little quiet. I hope you can hear me. So thank you to the organizers for letting me be part of this conference, and thank you for the kind introduction. I'm going to talk about how do we mine natural viral diversity for global health to design therapeutics and vaccines. Okay, so when we talk about chronic diseases, we typically think of non-communicable diseases like diabetes, cardiovascular diseases, and cancer. And then when we think about natural products, we often think about plant-derived or microbial metabolites. And the talk today is not about either of these, but rather viruses. Now, viruses are relevant because viral infections can lead to long-term health and immunity-related issues. For example, influenza can cause associations with cardiovascular risk, dengue immune activation, usually during secondary infection, and then coronavirus 2, long COVID, and some people are still suffering from this. And of course, you have chronic viral infections in the form of hepatitis B virus, hepatitis C virus, HIV, among many others. So viruses are relevant for this topic, and also viral proteins are actually natural bioactive molecules because they do have an important and powerful effect on human biology, and they are shaped by evolution, of course, so they do go through changes and result in new functions. They are exploring that biofunctional space. So it does make sense, it does make a lot of sense to analyze viruses, naturally occurring viral peptides sequences, to see if we can identify stable molecular targets or immutable targets for vaccines and therapeutics. These are sequences essentially that do not change or do not change much, and then they're very good therefore for drug design and vaccine design. Okay, keeping track on time. Okay, and of course, I think this is a well-known fact that there's a rapid growth in virus data. During COVID, there was a big example. We saw an explosion in virus data. These are just some stats. You have like 14 million sequences of nucleotides. You have about 64 million sequences for proteins. Now, these usually do not include, for example, the 17 million sequences that we have for SARS coronavirus on GSAID, roughly around that number. So, but in general, there's a lot of data publicly available, free for people to mine. Of course, you have to give the proper acknowledgement. And this is really a great opportunity for us to explore this space, to identify targets for, therapeutics and prophylactics. One big problem, of course, with viruses is the diversity. Viruses change quite rapidly, and in fact, there are two types of changes that we need to acknowledge. Number one is genetic diversity. This is change at the DNA level. Now, changes at the DNA level can impact diagnostic sensitivity and specificity because these assays, they recognize the DNA sequence, so changes there can impact. The other one is protein diversity. Now, not every change at the DNA level is going to result in a change at the amino acid level. So protein diversity is different from genetic diversity. And this can result in drug resistance because you have to have, your inhibitors blocking certain pockets, and if there's a change in those pockets, then your inhibitors are not going to work. But there's another side effect where these changes at the protein level can expand the host repertoire. So, for example, a virus that was only infecting, let's say, birds, because of these changes can infect humans. And we have seen this happen with flu and, of course, with coronavirus. This is also one of the big reasons that it was able to effectively infect humans. So that's zoonosis. And then antigenic diversity is basically the diversity from the perspective of the immune system. So you have recall, changes at the immune recognition level, and those changes can prevent the recognition by the immune system, and this is generally immune escape. Right? So there are these are some of the challenges, and again, focusing on conserved sequences is going to allow for us to overcome or at least mitigate and reduce these challenges. So conserved sequences, they are stable across strains, lineages, and time. That's why they're called conserved. They are observed to be like that because you need big data to make sure that this is a reliable observation. Small data may not be so reliable, and they often reflect essential functions, and that's why they cannot tolerate mutation, and therefore they remain conserved. These regions are therefore good targets for vaccines and therapeutics. Although on the flip side, it's an arms race. Evolutionarily, viruses have been observed to try to not reduce on purpose, but conserved sequences have been observed to be not very immunogenic, which helps the viruses. The diverse ones are more immunogenic, which is not good for these developments of vaccines and therapeutics. But it's not all that bad. Some of them are immunogenic, and then we need to think of creative ways to still use them and still get the best response, for example, by use of adjuvants. Okay. So these peptides improve robustness against viral evolution and escape. And of course, peptides are not that stable, so maybe you may have to use DNA technology in order to express them in the human body and then have the protein form produced for the purpose that you require. Okay, so focusing on conservation makes a lot of sense to reduce experimental and surveillance balance. Imagine if you don't do that, you design a vaccine and then you keep producing new vaccines. And this is what we see with influenza. There's like an annual influenza vaccine that needs to be generated because the viruses keep changing. Of course, there's some viruses, conserved sequences are limited, but there's a lot of effort and study that is being carried out to see how we can identify these conserved sequences and still be able to use them, even though the viruses look like they're changing a lot. And we've seen even HIV, for example, it's known to be very plastic disease, still does have some conserved sequences. Okay, and therefore it's very important to study viruses to find these conserved sequences, and we need a very good systematic approach to studying these viruses. So the study of viruses, there are two types of approaches, alignment dependent and alignment independent. I'm going to cover the alignment independent first because most of the studies are alignment dependent. Now, alignment independent, there are few approaches out there. We have our own approach. Here's an example of what we are doing to be able to carry out this. For example, you had three sequences up here that are very different from each other. Well, not very different, but different from each other. We call them unique sequences. And you can see the mutations, where the mutations happen. This is just a consensus, and these are the variable residues. So here are here NFF, so they're identical but different here. And here Q and L and Q. And overall, they're different. Now, if you were to break these sequences down into peptides, now peptides, why? Because peptides is what the immune system recognizes. 9 amino acid length is like a typical length of this recognition. So let's take 9 as an example. And we break it down into this repertoire of 9 mers or monomers. And then you have this list. And you notice from this list that all these ones are identical to each other and all these ones are identical to each other. And you do have some that are identical to all the three. So given this fact, it is possible then that you probably can capture all this repertoire with just 2 sequences instead of 3. So you have some sort of a reduction in the number of sequences. So this independent approach essentially does this kind of analysis and reduces your large number of sequences to smaller number of sequences. So for example, in this case, this middle sequence, this middle sequence becomes redundant. It's antigenically redundant. For the purpose of the immune system, it doesn't offer anything new. So we can remove this and only focus on studying the other 2. Right? And now this is an example for 3 sequences. Imagine you have millions of sequences. This approach can really reduce your data set to much smaller. And then it makes it easier for you to then study further, which then could be at an independent, sorry, the alignment dependent way. Now, one of the reasons why we do alignment independent is because you are not able to align them. For example, if you study within a species, sure, but if you study across multiple species, then alignment breaks down. So when alignment breaks down, or if you were to take, for example, all viruses, you're not going to be able to align them. They're different species. But this allows you to study them, reduce the number, and then think of strategies of how you can identify. Maybe then you can do species by species, or maybe then you can do, more like localized alignment, focusing on areas that you know are conserved functionally, and then so I know this doesn't completely give you conserved sequences, but it's a way to reduce the data set so that becomes more manageable. And then from there on, you could look into strategies to come up with conserved sequences, right? So now here's just an example. We took all viral sequences, protein sequences, almost 5 million. First, we removed duplicates, and this itself reduced the number almost half, because there's a lot of duplication in protein sequences. Not the same sequence, input twice, but a protein in different virus strains. Overall, the viruses are different, but the individual proteins might be identical because the difference might not be in this protein, but somewhere else. So because of that, you have duplication, and then you reduce it, you get the unique sequences, then you do what the approach that I showed you earlier, using our tool called unique mean to reduce it, and then you will get about 1 million in this. We got about 1 million. So from 4.9 million or 5 million, we reduced to 1.3 million through these two different steps. I think that's a huge reduction, and almost 74%, and then you have a much smaller data set to work with, and that should be able to help you and study and look for conserved sequences. So this is one approach. We published a lot. We even have a tutorial if you want to do this. We have the code. We make the code available. There's a tool. There's a GitHub page. You can look into that and download. Moving on, the alignment dependent approach. So we also have quite a number of publications on this you can look into. Generally, it's broken down into three parts. You download the data, you process the data, then you analyze the data. So you can look at all these steps here. I'm not going to go into the detail in the interest of time, but that's the general framework. Here's an example of the steps for the analysis. Let's say data collection is done, processing is done. Now we go into analysis. For the analysis, you need to have an alignment. Once you clean your data, you have an alignment. Here's an example of an alignment. And what we do is we do an overlapping peptide analysis. You choose a length. In this case, we choose 9 because that's typical length of recognition by the immune system. So you have like 1 to 9 amino acid, 2 to 10, 3 to 11. You kind of extract each one of those, and then you analyze the diversity within each one of those to see what's really going on. And again, the reason why we chose 9 is because this is an example of the peptide being nice, and the typical length is 9. Now, one of the things we do is we use Shannon's entropy to calculate the overall diversity within the block of 9. I'm not going to go into the formula, but generally there are two components to this formula. For each one of those blocks, we have multiple peptides. What we do is we see how many different peptides are there, and for each peptide we see what's the frequency of each peptide. So these are the two metrics that I use to calculate. Here's just an example. For example, we have this many different, and these are the frequencies of each. So all this is fed into the formula, and we get a number called Shannon's entropy number. The higher the number, the more diverse. The lower the number, the less diverse. So 0 is ideal, like 100% conservation. There's only one sequence there found in all the sequences. So 100% conservation. Okay, so it's affected by unique peptides and the probability of each. We actually have a tool we have built, the team had built, and you can use this to calculate the entropy. And then actually we expanded the work further, and we said actually within the block there's something going on. There's a population of sequences that we wanted to classify and better understand. So if you take one of these blocks, and there are, for example, this sequence is found 8 times. This one is found 5 times, this one is found, twice, this one once, twice, and then one. So we said, okay, can we classify them in a way so we recognize what's going on? So this one that's found 8 times, it's called index sequence. Now, index is like your wild type. So everybody else that's different from the wild type and of lower frequency, we call them variants. So all of these are variants. The variants can be classified further. You have major variant. This is like close competition to the index, to the wild type. Still variant, but very close. Can be close, can be not close, but yeah, it's like the dominant variant. Then we have something called minor variants. The difference between the way to understand minor is definitely they're not the major. And there's also another category called unique, which are only seen once and then they disappear. This could be sequencing artifacts, or this could be real ones, just didn't get sequenced again or just appeared once, we do not know. But in the data set, they only appear once. So minor are basically they appear more than once, but they are not the major. Okay, monotypes is just counting how many different variants you have. So you have all these terms, and these terms can help us better understand, dissect, and understand the dynamics of variation happening within each block and across the blocks. We actually built a tool, DIMA, available publicly for free, that you can upload an alignment and it will give you all these calculations. It can even handle metadata. There's a tutorial available. I don't have time to go through, but this is something that's available for people to use. And I highly recommend that if you have a sequence data, you want to look for conservation, you want to look at the analysis that I mentioned earlier, this is highly, and it also can do entropy that this one was doing, it can also handle that. So I highly recommend that you utilize this. You just got published recently. This is an example of how when you do the entropy, you can see the diversity. Let's say this is dengue virus 1, 2, 3, 4, and this is all of them put together. When you look at each one of them, you say, oh, there's a lot of conservation. But when you look at all of them together, you see the conservation is very little. These peaks, triangle peaks tell you where the conservation possibly are. And actually, we can give you an example. Even in those areas where you see conservation, you have to be careful. This is where the motifs are very useful. So let's say we take this position here. It generally looks conserved, some of them not so much. If we dive in one of these positions and we see something. So here's an example for one of the positions. You have all these sequences, and in dengue 1, the entropy is low, entropy is a little higher in dengue 2, very low in dengue 3, very low, 0, perfect in dengue 4. But when you put all together, it's very high. Why is that happening? Because in dengue 1, it's very conserved, 93% this peptide. This one, 72 and 27, so it's a bit of a competition from this peptide, and this one is very conserved, even more conserved. Because of this competition, you have kind of quite a number of dominant peptides. Now, the most dominant is 31, 38. So this is now the overall frequency. So you can see, it changes, the frequency changes when you consider everything, and as a result, the entropy is very high. Here's another one. There's another one where you can see dengue 1 entropy is very, very high. That's not very good. This one is very high also. This one is good. This one is very good, but overall it's not good. Okay, so here's another one. Entropy more than 1. It's usually above 0.5. It's considered quite diverse. So this is not good, this is good, this is not good, this is good. Overall, it's very good. So what are we really looking for? We want something like this. Entropy of dengue 1, very good. 100%. There's only one peptide. This one is very small, so that's also excellent because 1.54%, that's almost negligible. And then you have this one. So this is the kind of position that you want. So just look, look for conserve, and that's it. Sometimes it's not that easy to find conserve. You need these kind of tools to help you understand what's going on, and especially when you have different groups you have to deal with. So we found 44 in dengue that were like very low entropy, and they were only covering 15% of the proteome, and they were spread out. So again, not such an easy way to align and find them visually. No, you got to use tools. Yeah, there's those 44 sequences and the conservation within each. And there were 34 of them were conserved in 95% of the reported sequences of each serotype, which is really great. The lowest was around 80%. So if you use DEMA, you can actually set these percentages of conservation, and it will be able to check for you and tell you how many sequences you have. Now, these conserved sequences 44, we actually checked 24 months later whether there's any change in terms of the conservation level. 24 months later, no change. 38 months later, no change. Data increased, but still no change, which means there's something functional probably that's keeping these sequences relatively conserved. And we checked databases of functions, and we did find these sequences are actually involved. In fact, 27 of them are known to be relevant for structure and function. Then we also checked, are they important for immune response? There's no point in them being important for conserve, but they don't, trigger any response. So we checked that. We first used prediction tools, and we did find that quite a majority of them do trigger a response, right? And we checked for actually supertype. I don't have time to explain supertype, but just consider that, we checked for trigger of immune response, cellular immune response, and we found prediction results that supported. But we didn't stop there. We actually also did transgenic mice studies, and we found not for all the alleles of HLA, but some, and we found that there were some positive responses. So that's also very positive news. Then we also checked there's an existing database that actually has human T-cell epitope information. This is called IEDD, Immune Epitope Database. We checked to see if some of these sequences have been reported to trigger human response, and we found quite a number of them. So overall, it was very positive and encouraging that these sequences we found are immune relevant and can be used for this design. There's one other item that I just wanted to highlight, that conservation, you got to be careful. Conservation within your species is good, but if conservation extends to other species, it can be good, can be bad. For example, in the case of dengue, if your conservation extends to other viruses, but in the other virus, it's there, but not very conserved, that could be a problem. We call this antigenic sin, and it creates a confusion to the immune system in terms of recognition and can cause immunopathology. It can actually result in killing, damaging your own body. So here's an example. The 44 that we found, 27 of them are shared with as many as 64 flaviviruses. That's quite a lot of them sharing with a lot of viruses. So how do we understand that? And these are some of the biggest culprits, like Zika virus has exactly the same sequences as dengue, 22 of those. But is it highly conserved in Zika? We know in dengue is highly conserved, but in Zika, is it also highly conserved? That's the question. If it is highly conserved, maybe that's great, but what if it's not? So we dive in and we check, like this sequence, the first one found in 19 species, and St. Louis encephalitis virus is 97% conserved from 77 sequences. And that's maybe good. Very high, very high. So generally looks good, but we don't have data for these, so we do not know what's the situation. So if there's co-circulation, we want to design a drug or vaccine, let's say, and in that area you have 2 or 3 viruses circulating, then that could be a problem if the conservation is not high for those. Let's take an example, like this one. It's very high for West Nile, but Japanese encephalitis is less than 1%. And when we checked with 268 sequences, less than 1% added. So it's not conserved in that virus, even though it's found. So if you were to use this, and this virus also co-circulates and infects, and it can create a problem. So this is something also to be mindful about. You could play safe and say, oh, I will ignore anything that's found in another virus, but you might be losing out. That's why actually those are very conserved in other viruses. At the end of the day, you have all this information at your disposal, and you can better design your drugs to see what to target, and the rest, the other talks that we have covered about docking simulations and all that to identify the best candidate. So key summary, viruses are complex, but they do offer a tremendous opportunity to look for natural bioactive peptides. conservation is a good idea to focus for these bioactive peptides, but we must be mindful about the conservation going beyond to other related viruses that might share those sequences. My suggestion will be to focus on your species of interest, specific conservation, right, to avoid. But if you can manage analyzing the other ones and you see good conservation, then maybe you can still use it. The publications that we have done provide good data for you to look into this further and better understand. We also have established methodology and tools for this kind of work to be automated, so you can benefit from the effort that we have put in. Just want to thank everyone who kind of contributed to various publications that I cited here, mentioned here. Thank you very much again to the host. I hope I didn't exceed the time. Thank you for the opportunity. Unfortunately, I'm not going to be available for any questions, but you have my email. You can reach out to me if you have any questions, and we are open to exploring possible collaborations. Thank you very much.
Thank you very much, Professor Asif Khan. Are you still with us right now?
Yes, I happen to be here.
All right. Thank you. And Professor Eflisa Ashari, are you still with us?
Yes, I'm still here.
Sounds great. Thank you. All right. Now we'll move on to the question and answer session. We open to 3 participants to deliver the question, please. Number 1 here. Is there any other participant? Please mention your name and institution.
Yeah, my name is Indah Trilestari from Universitas Darussalam Gontor. I want to ask Prof. Siti Efliza Ashari. Actually, I'm interested in research of kubosome, but I want like to know if their costs are much different from compared to regular nanoparticle if the goal is commercialization. Thank you.
You want directly to answer? Yes, please.
Okay. All right. Perhaps I can I see the participants from that background, is it? Because right now I can see Dr. Bibit. So that is okay for if it is very difficult to change to the participant side. Because I want to see the students who ask on me.
Can we
it's okay if it's not. Okay. For the cubosome, thank you very much for the one who asking me the question. So cubosome, and right now I'm using NLC. So NLC and cubosome is a same. I mean, it's a one type of drug delivery system. But then, in terms of cubosome, the most important part is actually we want to make sure that the structure should be in cubic rather than NLC. Yeah, we can see only the spherical shape. It's very easy for us to do some analysis if the structure is in spherical. So it's very, there is no specific requirement to check on the structure. But then in terms of cubosome, yeah, the difference is we can use, because right now NLC we have a solid lipid and liquid lipid, but then for cubosome we have hydrophilic, same like we have to choose the correct hydrophobic and hydrophilic liquid, and also we can also tailor with amphiphilic drugs. So meaning that you have to check clearly your ingredient, and it must be suited to make sure that at the end of the formulation it can becomes to cubic structure. So I think that is the most important part. We have to make sure that the structure, because we are chemistry here, so we want to check the active site whether it can interact with the active ingredient. So you have to check through your active, your API, with the selections of your hydrophilic and hydrophobic, and also it can be used as amphiphilic drugs. Okay, but the most important part the most difficult task in cubosome is when you want to produce in a large scale. So it's very difficult for us to produce because you have to play around with the viscosity. So viscosity is very crucial part for producing the good and stable production of formulation. So for your information, L'Oreal cosmetic, usually they use that types of drug delivery system, cubosome. If you're familiar with the L'Oreal product, so that is they use the product in cubosome because they produce in large scale, but they can. I think the ingredient they use is suitable for the end product to be come out with the cubic structure. But NLC is actually based on different purpose. If you're looking for the cosmetic, usually they're looking forwards on nano emulsions and also the cubosome. But then on this specific targeted disease, I would like to treat the wound healing. So we prefer in NLC conditions. Yeah, I hope that I can answer your question clearly.
Does that answer your question? Okay, thank you. Please, any other question? Okay, silakan. Dr. Marlin.
Thank you, Professor Eliza, for sharing.
Now I can see the participants.
Oh yeah, yeah. The data was nice, like especially when we saw the fibroblast is able to close the gap in the migration assay, as well as in the in vivo data that showing the closure of the wound experiment. I was wondering, like, what is the explanation or what is the mechanism why this extract is able to improve the migration of that fibroblast? Like, what is it done to the cells so that they can move compared to the control one? And then the second one is, I was wondering, by making this NLS, why it act better? Is it because of the improvement of the solubility of the drugs, or because it can improve like the delivery of this drug inside the cell? I was wondering the mechanism why it's better using your NLS. Thank you.
All right. Okay, thank you very much. In terms when we are looking for the stability, so my concern is on its ingredients. So if you look at the mechanism, so the copritol that we use and also the oleic acid we use, liquid and solid lipids. It's actually the selection of that kind of ingredients. It's actually give improvement in terms of its stability, because you have to check through you have to make sure that the formulation should be stable enough for you to go further on the cell. So by right, if you ask me on the cell migrations, because in the first place I just show with you on the scratch assay. So that is the first preliminary work on the cell, where we use 3T3 fibroblast cell, which is a very simple explanation to us as a chemist. So when we see by using that with the cell, the cell is good migration with our system, I mean with our end formulation. So that's the reason why we can have a good migration with good closure region of the wound created. So I think because of the formulation itself, that it helps on the cell migration for the wound treatment. So that is the first one, and I mean that one is the second explanation. The first one is in terms of what? I forgot to take a note. Is it with the cell migration, right? You're talking about how it's work, the mechanism, if I'm not mistaken.
Do you still remember the last question, Marlin?
I think because of the cell migration. I mean, the explanation on the cell migration towards my mechanism of NLC. Is it?
The second one was like the NLC was aiming to enter the membrane cells. I was wondering, like, why it's active? Is it because of they are able to enter the cell membrane, or they just simply improve the solubility of the active ingredients? I was wondering that things, or both of them. Yeah.
So that is actually in terms of the cell entrapment. I mean, the cell entrapment works well with the formulation. So in terms of stability and the work performance by the cell migration. So I think it works related to the end formulation and with the cell itself, which can improve the entrapment of each other. All right? Okay, thank you.
Thank you. Maybe the next question, please. Oh, yeah.
Hello, this is Dr. Jitendra Patel from DMIHER India. Thank you, ma'am, for sharing your work. You mentioned in your talk, like in this plant, alkaloid, flavonoid, and terpenoids are there. So for wound healing, which compound are responsible or they are working together? That is my first question. And second question, you worked on NLC and NLC hydrogel, but you got the good result in NLC hydrogel. So what may be the reason that NLC only is not giving that much of wound healing? Thank you.
All right, thank you very much. The first one, the first question, in plant itself, in kratom itself, we have a lot of compound, active compound. So basically, as I mentioned earlier, the crucial, the prominent characteristics when we have the API which having an antioxidant, anti-inflammatory, and anti-angiogenesis in order to treat the compound. So by looking at the standardized crude extract that I presented before using LCMS, so the most important part is the mitragynine. And mitragynine, if you look at the structure, so it actually contribute to the improvement of the wound treatment. Because as we check through MS alone, so that will be that the proof, the evidence still give a good analysis for the wound treatment. I mean, for example, for wound scratching, but then the percentage is quite low, but then still there is a characteristics on wound healing. So mitragynine and also the part of the alkaloid, so because that one having antioxidant. All right. So in the second question, because of the NLC, you are talking about NLC and the crude extract towards the
hydrogel, NLC.
Dehydrate, yes.
NLC hydrogel.
Good question. All right, so I forgot to mention in this slide. So beside NLC and mitragynine, we also put the cooling agent, which is we are using sodium alginate. So that sodium alginate actually helps us to improvise the whole formulation. For example, as I mentioned earlier, it can having a cooling effect. That's why we need to have in the form of gel, because our purpose right now, if we have MS and NLC, we can just form in a solution like a serum, like a moisturizer. But our potential product, we are looking forward into a film, which can have a good behavior, and it can good for the external use for the transdermal drug delivery in order to treat the wound healing. So we are looking forward more on in a patch system film rather than in solid in a solution.
Thank you.
All right.
Thank you. Maybe next question. All right. No more? Okay, I think we can wrap up the session today. Ladies and gentlemen, what an exceptional session this has been. Today we have heard from 4 distinguished experts who have shared groundbreaking insight across biotechnology, artificial intelligence, pharmaceutical innovation, and bioinformatics. Together, this presentation revealed a powerful truth: the future of science depends on interdisciplinary collaboration, ethical practice, and a commitment to sustainability and education. Each speaker has shown us that innovation is not just about technology, it is about thoughtful, responsible efforts to improve human health and society. Once again, thank you very much for all invited speakers, Professor Asari and Professor Khan, for meaningful thoughts, and thank you for all participants for today. Hope everyone have a wonderful day. Thank you. I return the time to Master Ceremony. Thank you.
Thank you, Doctor. Thank you, Dr. Vivitri. Please give a round of applause for this insightful topics. We would like to express our appreciation for your active participation through this morning's programs. Lunch has been prepared and is now ready to be served. Please be informed that the next session will be began at 1 p.m. So we encourage everyone to return to the auditorium on time. Once again, enjoy your lunch, and we look forward to seeing you back for the next session. Saya informasikan kembali untuk makan siangnya sudah siap. Nanti setelah makan siang akan ada sesi paralel dan dimohon untuk tetap kembali ke auditorium jam pukul 1 siang. Terima kasih. For all the participants, please come to the stage to take a photo session together. Untuk para peserta hari ini dimohon untuk maju ke depan untuk melakukan sesi foto bersama.
gatel.
Gatal karena infeksi jamur? Pakai krim 88. Krim 88 tidak lengket dan cepat meresap. Dengan sensasi dingin dan aroma yang fresh, krim 88 kupas tuntas infeksi jamur.
Pakai luka gel aja.
Luka gel, cegah infeksi pada luka tanpa noda. Bunuh 99,99% bakteri dalam 30 detik. Luka gel, aksi cepat antiseptik luka.
Gatal jamur selangkangan, krim 88 solusinya. Tidak lengket dengan sensasi dingin dan aroma yang fresh. Semua tuntas pakai krim 88.
pukul 1.15. Dimohon untuk tetap berada di tempat dan mempersiapkan diri.
Gatal jamur selangkangan, krim 88 solusinya. Tidak lengket dengan sensasi dingin dan aroma yang fresh. Semua tuntas pakai krim 88. Mikaya. Gatal jamur selangkangan, Krim 88 solusinya. Tidak lengket dengan sensasi dingin dan aroma yang fresh.
Selangkangan semua tuntas pakai Krim 88.
Mikaya.
Gatal jamur selangkangan, Krim 88 solusinya. Tidak lengket dengan sensasi dingin dan aroma yang fresh.
Selangkangan semua tuntas pakai Krim 88.
Mikaya.
jamur selangkangan. Krim 88 solusinya. Tidak lengket dengan sensasi dingin dan aroma yang fresh.
Selangkangan semua tuntas pakai krim 88.
Mikaya.
Good evening, ladies and gentlemen. The parallel session will begin shortly. Please, maybe be seated. Good afternoon, ladies and gentlemen. Welcome back to the third International Conference on Natural Products and Chronic Diseases 2025. Natural Products for Quality of Life, Advancing Global Health and Sustainability. Ladies and gentlemen, we are now entering the parallel discussion for our conference. In this segment, participants will be divided into 5 rooms according to the topics and presentation schedules. Each session will be featured insightful discussion led by our speakers and moderators. The parallel session will be held in the 5 break rooms. Topic 1 in auditorium room, topic 2 in meeting cyber room, topic 3 in exhibition room, topic 4 in cyber room 1, and topic 5 in cyber room 2. Thank you for your participation. You might now process to your respective rooms. We invite to Dr. Vivitri Dewi Prasasti, MSc, to be our moderator in this parallel discussion. To Dr. Vivitri, the floor is yours.
Good afternoon, everyone. Assalamualaikum warahmatullahi wabarakatuh. We now continue to parallel session. Let me lead for this auditorium. We have 7 presenters today, but we will divide into 2 sessions. First, we will invite 4 first presenters. Maybe I would like to invite first Aisha Siddiqui. Please welcome. And second one is Rachel V. Parkon. Please come board. And third one, Dr. Jitendra Patel. Please come forward. Yes, please. Please welcome also Windy Chiang. Windy? Okay, please come forward. Thank you. So before we start, let me share about the guidelines. So we will have 10 minutes presentation for all of you, but that's include Q&A. So hopefully you guys can present up to 8 minutes, and then continue 2 minutes for discussion session. All right? Okay. So now we invite Ayesha Siddiq for first giving presentation with title Geographic Influence on the Phytochemical Composition of Heptisuvalence Aerial Parts. Time is yours. Okay, it works.
Hello. Assalamu alaikum warahmatullahi wabarakatuh. Good afternoon, respected professor, researchers, and my dear colleague. Myself, Ayesha Siddiqui from Kumaon University, Nainital, India. It is honor to me to represent my research work on the title of Geographical and Climatic Impact on the Phytochemical Compositions and Biological Activities of Heptisuvalence Point, under the supervision of my supervisor, Professor Geeta Tiwari ma'am. Please next. Now come to my contents which I used in my presentations. First is introduction, second material and method, result and discussion, significance of the study, key finding, conclusion, references, and last one, acknowledgement. Please next. Please next. Now come to first introductions, why Heptisuvalence point plant? Because its habitat is tropical, it's generally found in tropical, subtropical region, roadside, wasteland, and it's found 1200 meter as a altitude. Second main reason is major bioactive compound, that is sabinein, E-caryophyllene, beta-pinene, 1,8-cineol. Third point, therapeutic potential. It have antimicrobial, antioxidant, anti-inflammatory, and last one, insect repellent properties. Traditional uses of my plant, that is, it is used in medicine for treating wounds, fever, digestive issues, according to Devi et al. And Sharma et al. Please next. Classifications of my plant, that is, kingdom is Plantae, class Menglopsida, phylum Tracheophyta, family Lemmiales, order Lemmiales, family Lemmiaceae, genus Heptis. Please next. Descriptions of Heptisuvalence plant, that is, its stem is quadrangular, branching 1-2 meter tall, leaves is oblate, lanceolate, and root is taproot with lateral root, and seed is tiny, oval, 1-2.5 mm long. Please next. Vernacular names of Heptisuvalence, that is, in Marathi is called Jangli Tulas, Drab Tulas, in Malayalam Natapucheri in English, American mint, bush mint, in Hindi, Vilayati tulsi, in Sanskrit, bush tanna, in Assamese, Tokma tita. Please next. Sampling site. That is my sampling site where I collect my plants in 4 different locations. First is Pithoragarh, HSP. Second one, Rishikesh, is HSR. Third one, Bisalpur, HSB. Last one, Pithoragarh, HSP. Last one is Mohonia, HSM. Please next. Geographical details. First, my plant location HSM, that is 25.16 north, 83.62 east. Second location, that is HSP, are 29.60 north, 80.19 east. Third is HSB, its coordinates are 27.23 north and 78.01 east. Last location HSR, its coordinates are 30.13 north and 78.32 east. Next. Now come to methodology of my research work. First is survey and sample collections. First we survey in different locations. After it we collect my plants. Second step is environmental analysis. When we collect my plant, then also we record my all environmental conditions. Third step is oil extractions. Oil extractions is done by hydrodistillation process. Fourth step is phytochemical analysis. It is done by GC and GCMS. Fifth step is soil analysis. Soil analysis for pH, nutrients and growth impact. Last is data analysis. After, at the last step, we used establish relationship between variables. Next, please. Essential oil extractions process. Oil extractions is done by hydrodistillations for using Clevenger type apparatus. Second is drying and storage. For removing moisture, we used anhydrous sodium sulfate and it's stored at 4 degrees Celsius in BOD, biological oxygen demand. Last one, yield calculations. We calculate yield of oil which I contain by hydrodistillation process. Next one. In this slide, we show my all methodology. These are my collection site, hydrodistillation process, and last one is my oil, which I contain in vials. Please next. These are my GC and GCMS conditions, which I used in my sample oil. Please next. Essential oil yield and chemotype diversity. In HSR found highest yield at 0.11%, and in other side we found 0.10% in HSP, HSB, and last one is HSM, which you clearly see in this side. And in GC and GCMS we found 50 to 60 compounds. Please next. Previous literature review we show, according to Mel et al., its report 1.2 volume by weight yield from Hyptisavlon. Second, Olivier, which is report that highest yield 0.85% weight by weight. And last one is Nabi et al. It's reported very low extraction oil yield, that is 0.75% by Hyptisavlon plant. Please next. These are my GC chromatogram, which you clearly see In HSB, sabinein, E-caryophyllene, Z-alpha, bergamotol, abita, 8-12-diene, 8-13, beta-abitol, 18-ol. In HSB, you clearly see E-caryophyllene is major compound. In HSR, sabinein, terpinolene, 1,8-cineol, E-caryophyllene, abita, 8-12-diene. In HSM, sabinein, 1,8-cineol, E-caryophyllene, terpinolene, biogermacidine, sclerine. In this HSM collection site, we clearly see sabinein is the major compound. Last one, HSM, sabinein is the major compound. Please, next. These are all my major compound which I identify by GC and GCMS. Please, next. Chemotype profile Rishikesh HSR. Sabinein found 33.8%, which is highest major compound in Rishikesh collection site. Please, next. Chemotype profile Mohonia and Pithoragar. In this slide, you can clearly see caryophyllene 16.9% is major compound, and in Pithoragar region, caryophyllene 18.7 is the major compound except other compound. Please, next. My last site is Bisalpur, HSB. In HSB, sabinein found 15.7 and caryophyllene same 15.7% compound found. Please, next. Diverse climatic observation across sample site. These are my environmental data which I used in my research work. I take high temperature, low temperature, average temperature, sunshine hours, rainfall, rain days, pressure. Please, next. Soil parameters and their influence. In this slide, we mention soil pH, EC, organic carbon, nitrogen, phosphorus, potassium, sulfur, zinc, boron, iron, manganese, and last one, copper. Please, next. Micronutrients, dynamic and ecological implications. First is iron concentrations found measured in HSM collection site, which is 59.6 mg per kg. Second is zinc concentration, which is highest in HSB, and boron range is found 0.07 mg per kg in HSM and 0.81 mg kg in HSR. Last is copper abundance is found in HSR, which is 2.9 mg per kg. Please, next. Significance of my research work, that is understanding chemodiversity. Second is optimizing harvesting time. My research work is necessary for optimizing harvesting time found. It is best and condition for optimal therapeutic potentials. Third is breaching research gap. And last one, potential for drug delivery. It is most important for pharmaceuticals, cosmetic, and herbal formulations. Next slide, please. Conclusion of my research work. First is environmental factors. Environmental factors are crucial in driving chemotype diversity. Second conclusion of my research work, that is industrial and therapeutic uses, which is chemotype diversity has significant implications for potential industrial and therapeutic uses. Last one, further study of my research work, that is further study are recommended to explore seasonal variations. Please next. These are my references which I used in my presentations. Please next. Acknowledgement. I acknowledge to Professor Chitra Pandey, ma'am, Dean Faculty of Science, Head Department of Chemistry, DSB Campus, Nainital. Second is Professor Geeta Tiwari, ma'am, my supervisor, Nainital, for continuous support and guidance. Third is, I sincerely acknowledge the financial support I have received from DST Inspire, New Delhi, Indian government. Last one is, I would like to extend my thanks to my lab mates, Abha, Manisha Joshi, Shreya Jokaria. Thank you.
Thank you. Before we proceed the Q&A session, now we continue to next presenter with Ms. Maria Rachel Parkon with her title Extraction and Microencapsulation of Bromelain from Philippine Ananas Comosus Peels, Safety Evaluation in Tropical Application. Time is yours.
The Philippines is the leading producer of pineapple in Southeast Asia and has exported approximately 680,000 metric tons of pineapples in 2024. 20% of the pineapple plant is utilized as a viable product, while the remaining 80% composed of the crown, peel, leaves, and core, as well as the stem, is discarded, resulting in substantial environmental waste. These pineapple byproducts can be repurposed as potential sources of high-value bioactive substances, particularly enzymes such as the bromelain. Good afternoon, everyone. My name is Rachel from the Department of Science and Technology, Philippines, and I'll be presenting to you this afternoon our study. Next, please. Extraction and partial purification of bromelain from Philippine Ananas Comosus peels, safety in topical applications. Next, please. Bromelain is a mixture of proteolytic enzymes derived primarily from the fruit and stem of the pineapple plant, but it is also present in other parts such as the peels. Its natural enzyme-based mechanism provides an advantage over synthetic enzymes in topical product applications on top of its anti-aging, anti-inflammatory, and exfoliating properties. Next, please. However, bromelain production remains constrained due to its complex extraction and purification process to obtain high purity and good yield. To address these challenges, the aqueous two-phase system extraction has demonstrated significant potential for primary recovery operations in downstream protein processing. Next, please. The aqueous two-phase system is an eco-friendly and sustainable extraction procedure made up of either two incompatible polymers or a combination of a polymer such as polyethylene glycol and a salt such as magnesium sulfate. It is designed to remove major contaminants and concentrate the enzyme. In this system, you could see that the bromelain tends to migrate to the upper polymer phase, which is the upper phase, which has mild hydrating effect, interacting with proteins having moderate hydrophobicity, such as the bromelain. Next, please. Now, in this study, our group focused on obtaining bromelain from pineapple peels through aqueous two-phase system extraction and partial purification, and applied these enzymes in topical formulations. Next, please. Yeah. For the methodology of the study, fresh pineapple peels were obtained from a partner halal-certified manufacturing company in the Philippines. These peels were submitted to the laboratory for heavy metals analysis to ensure safety for use as a raw material. The cleaned peels were processed to obtain crude extract and subjected to aqueous two-phase system using polyethylene glycol and magnesium sulfate for partial purification. The extract was then analyzed for proteolytic activity following Murachi method. Next, please. Next, please. Next in the methodology include the freeze drying of the extract to obtain a powdered form of the bromelain, which is easier to handle for further applications and analyzed for its gelatin digesting unit assay. The bromelain powder was then applied in selected halal-compliant cosmetic formulations and subsequently analyzed for microbial profile and human repeat insult patch test to establish safety for use of the cosmetic products. Next, please. Encapsulation of the extract with gum arabic and maltodextrin was also conducted. This is to determine its feasibility with the bromelain extract. Encapsulation can significantly influence stability, physical characteristics, and enzymatic performance of the resulting powder for future applications. Next, please. Next, please. Yeah. For the results, this table shows that the pineapple peel used for this study was suitable as a raw material as it passed the limits set by WHO FAO for heavy metals in fruits, as well as the ASEAN limits for heavy metals in cosmetics, since the end product is a bromelain for cosmetics application. Next, please. This study explored multiple combinations of polymer PEG and salt magnesium sulfate to obtain optimum extraction condition. Here in this slide presented are the best results that we have obtained among the many trial combinations. Results indicate that the combination of 10% PEG and 30% magnesium sulfate obtained the significant yield and enzyme activity for bromelain's exfoliating properties at lower production cost compared to other combinations. Next, please. Bromelain powder has excellent solubility in water and was successfully incorporated into previously standardized formulations of gel cleansers and serum at 2 to 5% in the formulation with acceptable pH of 4.5 to 7 that supports enzyme stability and formulation compatibility. Next, please. For safety profiling, the cosmetics were analyzed for microbial analysis, and results show that the products with bromelain passed the ASEAN guidelines for limits of contaminants in cosmetics. Next, please. Furthermore, results of the human repeat insult patch test indicate that the products with bromelain are non-irritating and are generally safe to use for normal skin. For the attempt on encapsulation, Table 2 shows that encapsulated bromelain retained protein digesting activity at 372 GDU, almost equal to that of the non-encapsulated counterpart at 385 GDU, with an encapsulation efficiency of 96.7%. This indicates that aqueous two-phase system not only partially purifies bromelain, but also yields a stable starting material for encapsulation. The preserved activity confirms that the carriers that we used, gum arabic and maltodextrin, did not chemically interfere with the bromelain's active sites, but will act primarily as a physical barrier. Next slide. Yeah, today's skincare market relies heavily on chemical exfoliants, which often cause redness, peeling, and irritation. Bromelain offers a gentle, natural enzyme exfoliation, unlike harsh chemicals, making it suitable even for all types of skin, addressing the increasing demand for natural cosmetics. Please. In conclusion, pineapple peels are a suitable source of bromelain for cosmetic applications. The aqueous two-phase system enabled efficient and eco-friendly partial purification of bromelain. Bromelain-based formulations caused no irritation and presented low risk for hypersensitivity reactions. And as a recommendation, encapsulation of bromelain via freeze drying can significantly influence the stability, physical attributes, and enzymatic performance of this active compound for further applications in the future. Before I end my presentation, I would like to thank, next please, our agency, the Department of Science and Technology in the Philippines, the DOST PSHERD, my institute, the Industrial Technology Development Institute, our partner industries, the Hylas Marketing and Dole Philippines. And thank you for your kind attention. Terima kasih.
Thank you very much. Very interesting. Now we proceed for the third presenter. I would invite Dr. Jitendra Patel with the title Development, Optimization, and Physicochemical Characterization of Liposomal Formulations Containing Ethanolic Leaf Extract and Flavonoid-rich fraction of Albizia Lebec for Enhanced Hepatoprotective Delivery. Time is yours.
Good afternoon all. Myself, Dr. Jitendra Patel from Datta Meghe College of Pharmacy, Datta Meghe Institute of Higher Education and Research, Wardha, Maharashtra, India. Today I'm going to present into one of the important medicinal plant for the development, optimization and physicochemical characterization of flavonoid-rich fraction and even in the plant extract, how it can have the better drug delivery for the hepatoprotection. So when it comes to the particular plant, next please. This is the medicinal plant. As we know that liver illness that is the impacting globally, and there are many reasons which can impact the liver, maybe drug toxicity, maybe food toxicity. So this medicinal plant already it has been explored for its hepatoprotective activity. This is known as Sirish, that is one of the common name over there, and which has been used for a long time even in Ayurveda or in Unani system of medicine for its different, the huge pharmacological action. This plant is notably known for the presence of flavonoid, say for example camphorol, quercetin, glycosides, and due to the presence of these constituents, it gives the better antioxidant and even in hepatoprotective properties. Liposomal drug delivery system, why it has been decided, because it has not been explored before also, and whatever the plant constituent is responsible to give the hepatoprotective activity that are having poor solubility and maybe the less bioavailability. So next please. When we go for the liposomal formulations, we are addressing its solubility issue. Like if flavonoid will reach to the hepatocytes, it can have the better efficacy. So in the liposomal, the hydrophobic and hydrophilic, the two cores supposed to be there, and when the drug which is active pharmaceutical ingredient and responsible to give the hepatoprotection, it may have the better solubility and the better absorption. Next please. When we talk about the research gap, this plant, it already reported for the having hepatoprotective activity, but it has not been formulated in liposome, and moreover the flavonoid content are responsible to give the hepatoprotective. So when we go for such an advanced formulation, definitely we'll be able to address the solubility parameter as well as the bioavailability, which can further enhance the overall activity in terms of hepatoprotection. Next please. The main objective of this work is to formulate, optimize physicochemical characterize of liposomal preparation of the ethanolic leaf extract as well as the flavonoid rich fraction of Albizia labac, and assess their in vitro release, kinetic and short-term physical stability for the improved hepatoprotective administration. Next please. When we talk about the phytochemical content, if it is the normal extract, in ethanolic extract, the percentage of the yield, that is the 12.4, whereas in the flavonoid rich fraction, it is 4.8. So at last, when we talk about the total flavonoid content, which is our ultimate goal, so that the delivery will be more. So here we can see that is in the ethanolic extract it is 23.4, whereas it is 78.5 in flavonoid rich fraction. So the better yield we are able to get in this case. Next please. When we formulate into the liposomal formulation to the particular extract after having the flavonoid fraction, we need to adopt certain steps, like we have to add the different lipid parameters, surfactant, so that there will be the lipid bilayer which can have the proper encapsulation even for the flavonoid fraction and for the plant extract, so that we can assess its release parameter, its efficacy further. Next please. These are the parameters which has been selected for the formulations in the formulation variables in this optimized condition, like phospholipid concentration. It has been tested, and also in the optimized condition it has been taken in the first it has taken 5 to 25 milligram per ml, and in optimized we have taken the 20 milligram per ml. So in that way we have already tested for the particular profile, and we have reached to one goal, and optimized condition the formulation has been formulated. Next. When it comes to the particle size distribution, if you see the flavonoid loaded liposome, and in the in terms of ethanolic extract loaded, so the particle size it is 138.2 nanometer, whereas in ethanolic extract loaded liposome it was 152.4 nanometer. Next please. In the same, when we have gone for the zeta potential distribution, we have received that flavonoid loaded that was the 36.2 MB, and in ethanolic extract loaded liposome it was minus 32.6 MB. Next please. Encapsulation efficiency of extract loaded versus flavonoid loaded liposome. So in ethanolic extract loaded liposome the efficiency was 67.8, whereas in flavonoid rich fraction loaded liposome it was 82.5. Next please. When we have gone for the transmission electron microscopy for those particles which has been loaded into the liposome for the ethanolic extract and for the flavonoid rich fraction loaded in liposome, so we have received that the flavonoid rich fraction loaded liposome have given the uniform marginal reduced size, and which is very important for the even absorption and to show the efficacy. Next please. The FTI analysis also has been done for these two formulations, the ethanolic extract loaded liposome and for the flavonoid rich fraction loaded liposome. So in flavonoid rich fraction loaded liposome, you can see into the graph that it has given the better absorption band, which is the indication of the uploading of the more flavonoidal content, which may have the better efficacy later on. Next please. Drug release profile for the liposomal formulation for both the formulations. So the drug release profile, when we checked, it was the flavonoid enriched formulation exhibits superior cumulative release rate in compared to the normal crude extract loaded into the liposome. Next please. These are the profile in the 0 to 24 hour. In the extract loaded, it was 65.7, whereas in the flavonoid loaded at 24th hour, 82.3. So that is the indication that the release rate it is having time bond and in flavonoid. Next please. Stability data also has been assessed, and we have found that flavonoid loaded liposome have given the better efficacy, better stability during the fraction of time. Next please. Vesicle size also has been observed for 30 days, and it was having uniform vesicle size during the stability testing. Next please.
Excuse me, you have 2 minutes left.
Yeah, I'm going to conclude. Next slide. Next slide, please. Yeah, the present studies successfully developed and optimized the liposomal formulations containing the ethanolic leaf extract and the flavonoid-rich fraction of Albizia labac, demonstrating significant enhancement in the physicochemical characteristic and delivery performance. Among the two systems, the flavonoid liposome exhibited distinctly superior attributes, including higher encapsulation in terms of smaller size and all. The electron microscopy analysis confirmed that formulation of smooth spherical predominantly similar vesicles and validating the effectiveness of it. The flavonoid-loaded liposome also showed the more controlled and sustained release profile, supporting their potential and prolonged hepatoprotection activity. Overall, the finding of that liposome encapsulation significantly enhanced the stability, bioavailability, and the release behavior of Albizia labac flavonoid, underscoring their promise and advanced hepatoprotective delivery. Thank you.
Thank you, Dr. Jitendra. Now we move forward to the fourth presenter with Windy Chiang, with the title, The Bioactive Driver of Anti-Diabetic Activity in Avocado Leaf Aqueous Fraction Extract, a potent dual inhibitor of alpha-glucosidase and oxidative stress. Time is yours.
Good afternoon, Professor, Doctor, colleagues, friends, and everyone. My name is Windy Chiang from Pancasila University, and I'm today proud to present our collaborative research and partnership with National Research and Organization in Indonesia. And I want to give my gratitude to my supervisor, Professor Muhammad Hanafi. And today I'm going to present about the bioassay-guided identification in avocado leaf, a potent aqueous fraction dual targeting for anti-diabetic activity. Next, please. Well, we are motivated by a critical global health challenge. The International Diabetes Federation Diabetes Atlas show a recently rising prevalence of diabetes, underscoring the urgent need to effective and accessible manage for strategies. Next, please. And the key of therapeutic target for managing the postprandial glucose is in the enzyme called alpha-glucoside in the small intestine. It can be a provider on a crucial mechanism for blood sugar control. Next, please. This a little bit about the avocado leaf potential. In Indonesia, we have a lot of species from avocado, but the Persea americana male is one of the commonly found in Indonesia, and it's more than just a fruit. The leaf is documented in recent studies, show a remarkable multi-potential profile, including significant antioxidant, hepatoprotective, and notably the anti-diabetic potential, evidenced by blood glucose reduction and even pancreatic cell regeneration in animal studies. Next, please. And the research proposed. Therefore, our research aimed to scientifically validate this potential. We have two core objectives. First, to evaluate the antioxidant and alpha-glucosidase inhibition activity for an avocado leaf aqueous fraction, and secondly, to identify the key phytochemical compound responsible for this activity. Next, please. This is for my research methodology. We employ a structure, and we began with the ethanol extract, 70% of the leaves, followed by the fractionation to isolate the aqueous fraction, and this key fractionation was then rigorously evaluated for the alpha-glucoside inhibition and antioxidant capacity via DPPH and ABTS assay. Finally, we use the advanced LC-HRMS and NMR analysis to pinpoint the active compound. Next, please. The result were highly promising. The aqueous fraction shows the activist, the total phenolic and flavonoid content. Most importantly, It exhibits potent antioxidant activity with the very low IC50 values in both DPPH and IBTS assay. Crucially, it demonstrates a strong inhibition in alpha-glucoside inhibition, impressive with 9.16 ppm, indicating its direct antibiotic potential. Next, please. Furthermore, we will compare all the fraction, the aqueous fraction, and rank among the most potent from the all fraction, and it balanced the activity. Moving to the identification with NMR and LCH-RMS, conclusively identifies the major compound from this avocado. Next, please. Well, 7 carbon sugar alcohol with the molecular formula, carbon 7, hydrogen 16, and oxygen 7. This is what we found in avocado percentiamycanamyl, and this is a significant finding as the percetol is known as the compound now linked to our work and this specific dual bioactification. Next, please. Well, in conclusion, our bioassay guided study successful identify the avocado leaf cause the fraction and potent source of antioxidant and alpha-glucose inhibitor agent. We have identified that percetol and its major constituents and providing as its strong scientific basis for the traditional use of the avocado leaves, and the highlight its potential is a natural dual targeting indicate for the diabetic therapy. Thank you.
Thank you. Now we continue to discussion section. We open 3 questions. Please mention name and institution. Any question from the floor? Oh, please, Pak Ilya.
Hello, I have a question actually to our guest from Philippines. Do you see significant differences in bromelain content between different pineapple varieties? What is the variability in bromelain content between different varieties, if you know that?
Yeah, thank you so much for that question. We have not touched about the varieties of pineapples because we focus more on the Philippine variety of Ananas squamosus. But I believe there would be a difference in terms of their proteolytic activity depending on their source, particularly the influence of the environment, the soil where it's coming from, other contaminants that may be present that might affect the enzyme that is present in every pineapple variety. Thank you.
Thank you. Next question, please. Oh, please, Dr. Malin.
Thank you very much. My question is for the researcher from Philippines. It's nice study. I just know that the bromelain is very interesting. I was wondering if you do that extraction using the polyethylene glycol and MgSO4, magnesium sulfate, this is only bromelain that can go to that phase, or is there anything that also goes in that phase? And the second question will be, have you compared the activity of your bromelain that you isolate with the bromelain, for example, from Sigma? How it's the difference in terms of the activity? And do you have any comment in terms of the stability of the bromelain with that micro encapsulation? I was wondering, does it also experience heat, or I missed that part, whether it experienced the heat or it's just the freeze drying? Thank you.
All right. For your first question, I hope I'll be able to capture the three questions properly. Yeah, for the first question, I believe there are other components or compounds still present in our extract because the aqueous two-phase system only offers partial purification. This is so because what the aqueous two-phase system really do is to eliminate major contamination, major contaminants, and concentrate the enzyme. But there are other components since this is only a partial purification. Now, you would wonder, why did we push with partial purification, not a total purification of bromelain in this process? Our intention as a research and development institute in the Philippines is to transfer these technologies to the industry. We are hoping that someone from the industry would adopt the technology. There are techniques for total purification of these types of enzymes like chromatographic techniques, but when you transfer the technology to the industry, it will be very costly for them. There might be no industry that would be interested to adopt the technology. So we proceed with the partial purification and identify the proteolytic activity and the gelatin digestion unit assays such that we could establish also that there is really bromelain in our sample that will be used or useful for cosmetic application. Now, we have not gone through comparing our bromelain with that of a pure substance like from Sigma or other suppliers. That may be a very good way forward for us. But again, our intention really is to have this technology adopted by the industry. So we are good already with partial purification of the bromelain in pineapple peels. Anyway, the raw material is peels. So it is not that comparable when you get bromelain from the core, where there is really the concentration of bromelain, but it is a very good way to valorize these types of environmental waste. And then lastly, in terms of the microencapsulation, I just presented, because this is actually our way forward, the study on bromelain was completed already, but our way forward is what we are doing right now is the encapsulation. I included it in my presentation because I would like to present that there is a very good technique that researchers or the industry could do to protect this enzyme, which is the encapsulation using freeze-drying technique. There are also other techniques being used, but we use the freeze-drying technique because enzymes tend to depreciate when you subject them in high temperature like that. So we are proposing the freeze-drying technique for encapsulation. But again, this is a way forward. This is an ongoing study. I just presented it as part of what we have done already so far. Thank you. I hope I have answered your question. Thank you.
I think it's pretty clear, right? I was wondering, when you do the fractionation, how much percentage you got from the bromelain that you extract? Thank you. Next question, please, if there are any. Maybe I would like to ask question addressed to Asha Siddiqui. Since you compare the phytochemical composition from, I would say, American mint, right? How big different the phytochemical composition when you compare from different areas or geographic? You did the comparison from different plants that the American mint, right? The Hyptis cephalens. You check the composition of phytochemical compound. How big different the composition of each? Yeah. Comparison of phytochemicals composition in the different area. Because you do geographic influence.
some major compound like savinine, E-caryophyllene found major, and in one location that is HSM, which is located in Bihar, where found beta-germaserine, bicyclogermaserine, and in other state this compound is disappear. That is the major difference.
Thank you. Is there any question? Maybe my last question addressed to Dr. Jitendra. Since you did the development and optimization for analyzing the liposomal formulation containing this ethanolic leaf extract of Albizia lebec, and you apply it as a hepatoprotective delivery. Did you also check the toxicology for this application?
Not yet. Actually, this plant, it has been explored 10 years ago for having hepatoprotective activity in extract. So we have selected the leaf only because of the presence of huge number of flavonoid, and because of that only it was reported hepatoprotective activity. This time we are formulating it in liposomal encapsulation to enhance the solubility at the same time of bioavailability. So toxicity it has already tested 10 years ago. So in that way we are moving ahead. So we see that as compared to plant extract, this liposomal formulation, how efficiently it is going to give the hepatoprotection. Yeah, thank you.
Thank you very much. All right. No question more? Okay. I think we can wrap up for this session. Thank you so much for all four presenters. Give a big applause for all four presenters. Thank you. You may proceed. Thank you so much. Now we continue for the second session, inviting other three presenters. We invite Professor Sri Endarti Rahayu to come forward. Silakan Bu. Belum. Duduk dulu ya Bu. Thank you. And second presenter is Rizka Bella. Please come forward. Thank you. Thank you. And the third presenter will be Bunga Anggreni Sari, SSI M Biomed. Please come forward. Professor Sri Endarti Rahayu with her title Anti-Osteoporotic Activity of Pandanus tectorius seed extract in overectomized rats.
Okay, thank you, Dr. Vivitri. Good afternoon, everyone. Today I will present my research about this one. But it's not. Okay, my research about anti-osteoporotic activity of Pandanus stectorus seed extract in ovariectomas rats. Next, please. Females are more likely to suffer from osteoporosis than men. Osteoporosis and bone loss are both brought by imbalance between bone growth and Okay, thank you. Okay, so females are more likely to suffer from osteoporosis than men. Osteoporosis and bone loss are both brought by imbalance between bone growth and resorption caused by decrease of estrogen levels. According to a recent study, 200 million people worldwide suffer from osteoporosis. And 1 in 3 women over the age of 50 may be suffering it their lifetime. Osteoporosis mainly occurs in older female. 30% of women over the age of 50 are diagnosed during menopause because of sex hormone dysregulation leading to impaired bone remodeling. Next, please. Hormonal change during menopause that ultimately lead to the decrease in estrogen have a central role in this chronic disease development. In osteoporosis, the effect of osteoclasts are more active than these of osteoblasts. Therefore, bone is removed faster than its form. As a result, there is a severe of bone loss. Hormone replacement therapy is one of the most often utilized therapies for postmenopausal women at risk of osteoporosis. But this medication for long-term use may significantly affect human health, particularly in elderly population. There is the urgent need to create new medication to treat osteoporosis without adverse effect. Attention was paid to phytoestrogen implant to identify substance can provide the benefit of estrogen therapy without some adverse effect. Next, please. So, phytoestrogen are polyphenol compounds that are structurally similar to the steroid hormone. There are several plants contain phenolic compounds as well as have estrogenic effect that relieve menopausal symptoms. Potential alternative treatment for osteoporosis are the prophylactic and therapeutic effect of some plant-derived natural products, including scopine, Pandanus stectorus. So the studies aim to investigate the potential benefit of Pandanus stectorus seed extract on bone loss in the Everett-Thomas rat model of osteoporosis. So is the method. So after the rats were allowed to acclimate for 1 week, they were anesthetized and underwent resection to bilateral ovaries. A further 6 rats, the same group, also anesthetized, but only underwent resection of small sample of fat. So rather than the bilateral ovaries, no effects. All rats were monitored for 20 days before initiating the therapeutic regimen to allow them to recover from the operation. Next, please. So is the sick group. We divide to the sick group. The one group is the same group. It's not OVX. It's only give CMC 0.55% in 3 weeks. Okay, and the group 1 is negative control. We give CMC on 0.5% in 20 with OVX. This OVX, just we compare about the OVX and OVS with the treatment of seed extract or not, with the high dose, low dose, and medium dose. Okay, next please. See, okay, next please. This, okay, next. Okay, this is the treatment. Okay. Next, please. So the result, the phenolic content. Total phenolic content extract of Pandanus stectorus per evaluate. I was found that the extract of Pandanus dactylus seed contained total phenolic content at concentration 30.30 of gallic acid equivalent per gram. And its body weight, its body weight is not really influenced by the two body weight about the treatment. Next, please. Okay, the results show just a decrease in body weight growth on the third day of the administration of the Pandanus dactylus extract. It is natural reaction to adaptation of the new treatment, and this decrease occur in all treatment group. Successively, there was no significant difference actually in body weight. So from the body weight, it can be concluded the administration of the Pandanus dactylus seed extract at dose middle, low, and high didn't cause disturbance in body weight. Okay, next please. So the treatment is influenced to uterus weight. See, we can see here. Next please. Okay, so all groups that receive ovariotomy treatment show a significant decrease in uterus weight compared to the same without OVX. So this shows that the success of hormonal module action that will trigger the occurrence of osteoporosis in this set. So it's work. The research is okay. Next, please. So it is the analysis of bone trabecular density. We see here that the treatment is effect on trabecular density or not, because it's the characters that's important to show the treatment is effect or not on trabecular density. It's a good character to see the osteoporosis or not. Okay, next please. So only the positive control, so trabecular density was not significantly different with same. So it's only the positive control. But here that we see that the administration of C-Pandan has effectiveness in increasing trabecular density in osteoporosis rot. So it's worth the treatment. Okay, next please. So it's the pictures of the microphotography. Okay, so if proportion osteoclast and osteoblast. So if the treatment is work okay, so they will increase the osteoblast, not osteoclast. Okay, next please. So the number of osteoclasts could not be reduced. The same is impossible, but only the positive control group, so it's low, no significant difference. But increasing doses show that decreasing trend in proportion of osteoclasts to the number of osteoclasts. So the osteoclast is decreased, so it's work. So maintaining the bone mass relies on dynamic balance between osteoblast bone formation and resorption. So after menopause, bone resorption occurs earlier and bone mass is reduced. The trabecular bone microstructure is important factor actually to determine bone strength as well as bone physiology. The bone microstructure contours, especially trabecular microstructure, also have significant effect on the bone quality. Therefore, the evaluation of micro-CT, three-dimensional image, is very important for osteoporosis diagnosis. So estrogen deficiency induced by oxidative stress. Oxycodone can protect the cell from the damaging effect oxidative stress, and flavonoid through reduction of oxidative stress favor bone deposition, inhibit bone resorption, and regulating bone homeostasis. So flavonoid can preserve bone health by improving osteoblastogenesis and reduces osteoglycogenesis. So the result of the study show that Pandanus dactylus extract with high dose treatment could improve the loss of bone strength. Therefore, the Pandanus dactylus extract with high dose may be an attractive therapeutic agent for osteoporosis treatment. The conclusion, so the Pandanus dactylus extract with high dose restore of bone microarchitecture in ovariotomous rite, by encouraging the bone formation and inhibiting bone absorption. Pandanus dextrosid extract with high dose may be a new supplement for the osteoporosis prevention and treatment. So I would like to acknowledge the author are thankful to Ristek Brain Indonesia as fund grant were sanctioned under PDUPT or Penelitian Dasar Unggulan Perguruan Tinggi number 613 at 2021. Faculty and Biology and Agriculture, Fakultas Nasional, so that research work can be complete successfully. Thank you.
Thank you, Professor Endarti, for the insightful thoughts. Next, we will invite Rizka Bella for her title, Acute Oral Toxicity Study of Kebar Grass Ethanol Extract in Mice. Time is yours.
I'm Rizka Bella from Universitas Pakuan. It is a pleasure to be here today, and I'm honored to have the opportunity to present our recent finding conducted together with Apotheker and Rustiani and Farm titled Acute Oral Toxicity Study of Kebar Grass Pitum umbraculum well ethanol extract in mice mus musculus. Herbal derived products are becoming increasingly popular in worldwide, including in Indonesia. Kebar grass is a traditional medicinal plant widely distributed to Indonesia, especially in Papua. Used for a variety of therapeutic benefits such as antioxidant and fertility. Despite various studies on the reporting of pharmacological and phytochemical benefits, there are still limited scientific data on the safety profile and toxicity of the animal models. Acute toxicity testing plays a crucial role as an early step in safety assessment. It helps identify potential toxic symptoms, estimate the lethal dose, and determine whether a substance may cause harmful effect after a single high-dose exposure. This study was conducted following the 2022 Indonesian BPOM guideline for non-clinical in vivo toxicity testing, which provides standardized procedure for evaluating toxicity in animal models. Therefore, this study aimed to evaluate the acute oral toxicity of Biophytum umbraculum extract ethanol Kebar grass in mice to provide preliminary safety information for its potential development into pharmaceutical of therapeutic application. Next, please. This study used a fixed dose method with single oral dose of 300, 2,000, and 5,000 milligram per kilogram of body weight from Kebar grass ethanol extract and a negative control group receiving 0.5% NaCMC. A total of 22 female mice were divided into 4 groups, and acute toxicity tests were carried out in stages, starting with preliminary test, then main test, and limit test. Before administration and testing, the mice were acclimatized for 5 to 7 days. All animals are observed for 40 days to monitor clinical signs, mortality, body weight, and organ weight. All results are presented as mean plus standard deviation. Next, please. The result, all symptoms that occur up to a dose of 5,000 5,000 milligram per kilogram body weight do not result in clinically significant change. No further clinical signs were detected except mild transient grooming, which is considered a normal behavior response in mice were found after 40 days of administration. No mortality was recorded in any group, including control. At dose of 300, 2,000, and 5,000 milligram per kilogram body weight, the ethanol extract was well tolerated up to 5,000 with no observed lethal effect. The increase in body weight of mice indicate normal growth during in the study, did not indicate any significant physiology disturb. No significant difference in body weight were found between groups receiving the same treatment in individual dose. These findings are consistent with OECD guideline, which states the stable weight gain suggests absence of metabolic or toxic effect. The results support previous report indicating that herbal extract in high dose do not induce significant weight loss and may be considered safe from a toxicological perspective. Subsequently, from the observation of absolute weight and relative organ of the mice, there was no significant difference in the absolute weight of the liver, heart, lung, and kidney between control group and treated at dose of 300, 2,000, and 5,000 milligram per kilogram body weight. Also, overall, the finding of organ weight support the general safety of the extract. Although further subchronic tests with detailed histopathologies are recommended. Next, please. In conclusion, administration of ethanol extract of Biopitum umbraculum to female mice showed no sign of toxicity based on clinically observed behavioral change. Although a slight increase in average body weight was observed, this change was not statistically significant. There were also no significant difference in organ weight across treatment group. In the limit test, at the highest dose of 5,000 milligram per kilogram body weight, no mortality or significant toxic symptoms were observed. The result indicate that the ethanol extract of Biopitum umbraculum is relatively safe and dose up to 5,000 milligram per kilogram body weight, as it does not appear to induce ecotoxic effect in female mice. Next. Thank you.
Thank you very much. Very interesting. Next, we move on to the last presenter with Bunga Anggraini-Sari, SSC M Biomed, with her title, Anti-Diabetic Potential of Cryptophyllum fragrans DC leaves in silico phytochemical profiling, antioxidant activity, and molecular docking of LCMS compounds. Time is yours.
Oh, udah. Oke. Thank you, Dr. Vivit. Assalamualaikum warahmatullahi wabarakatuh.
Good evening, everyone. Today, I want explain about my research, the anti-diabetic potential of Kyrtofilum fragrans disease lives in silico phytochemical profiling, antioxidant activity, and molecular docking of LCMS compounds. This research is not only by me, but there is my promoter. There is Professor Dr. Edi Marsalim, and then there is Professor Dr. Muhammad Irsan Saleh, and the last there is Professor Dr. Ernawati Sinaga. So this is my outline. There is introduction, and then there is aim and study, and then there is material and methods, there is result, discussion, and conclusion. So we know that about diabetic mellitus is chronic disease, metabolic disorder characterized by hyperglycemia resulting from impaired insulin secretion, insulin resistance, or both. In Indonesia, the prevalence of diabetic continues to rise with substantial economic healthcare burdens. Medicinal plants have attract increasing interest as alternative therapeutic agents for diabetic, and Kyrtofilum fragrans are the synonym of Fragrilla fragrans, or in the local name there is a tembesu. Traditional use in Southeast Asia has shown anecdotal efficacy in managing diabetes, especially in Lingkiti district, Ogan Komering Ulu, Regency, South Sumatra. Next, please. Next, please. Okay. So, the aim of study to evaluate the antidiabetic potential of Kyrtofilum fragrans leaves through phytochemical antioxidant LCMS metabolite and in silico analysis target protein. Next, please. So this is the material and methods. The first one, use extraction material and process. And the second, there is phytochemical test and antioxidant, flavonoid, and phenols. And the third, there is LCMS compound, and the last is molecular docking. So the result from phytochemistry, there is alkanoid and flavonoid, steroid, saponin, and tannin. So this is the result from qualitative phytochemical. And this is the result from total phenol and flavonoid, which one phenolic compound, and this is the flavonoid compound. And the third, there is IC-50, and okay, next. Next, please. This is the result of LCMS compound. From the LCMS compound, we have 80 compounds identified, including glycoside flavonoid, iridoid, triterpenoid, phenolic, and terpenoid. This is by LCMS. So from the 80 compounds were identified in, we are molecular docking, and this is the 5 best result from molecular docking. This is the result from molecular docking. There is the A is luteolin, and the B is chloranthonin, and the third ferrolide, and the fourth stularin, and the fifth chichorin. And this is the discussion. So the ethnobotanical prevalence of Kyrtofilum fragrans supports in selection, but relies on single geographic source limit generation. And then a multidisciplinary approach combining phytochemistry, metabolite, profiling, and molecular docking. And the third, this integrative design highlight the potential of underexplored medicinal plants as anti-diabetic candidate. There is limitation, include absence of experimental validation, a single extraction method, and region-specific sampling. The future work should isolate lead compounds, validate in vitro or in vivo, and explore advanced delivery system. So the conclusion is Kyrtophyllum fragrans leaf extract demonstrate promising anti-diabetic potential supported by bioactive metabolite, and then the integrative analysis including phytochemistry LCMS profiling and molecular docking highlight compounds such as luteinizing with strong inhibitory activity against in DPP-4. This finding position Kyrtophyllum fragrans as a valuable source for developing anti-diabetic pharmaceutical. Further studies are required to isolate active continue, validate their efficacy through in vitro, in vivo, and clinical investigation. This is my reference, and thank you. I want to say thanks to Center for Botanical and Chronic Disease, my college, Universitas Sriwijaya, and Universitas Nasional. Thank you.
Thank you very much. Interesting study. Now we move on to the discussion session. We open 3 first questions. Please mention your name and institution. Yes, please. Mic for...
Good afternoon. My name is Francis Xavier from National Research and Innovation Agency, Republic of Indonesia. So I have a question for the third speaker that the receptor target for anti-diabetic chosen is DPP-4. My question is why use only select DPP-4 inhibitor as the specific target for docking? Have you screened other receptors or only choose DPP-4? And how about the positive control? Is the docking result of the LCMS compound is better than the positive controls, e.g. sitagliptin? Thank you.
Okay. Actually, in this research, I use three. There is DPP-4, resistin, and the last PPR gamma. But from the other, only DPP-4 the best answer. And I use positive control too. I use metformin and sitagliptin. Yeah, the same. The same, not better. Thank you.
Does that answer your question? Okay, thank you. Other question, please? Oh, yeah, Dr. Patel. Jitendra.
This is Dr. JP from India. My question is to Professor Sri. For osteoporosis, the plant PT which has been taken seed, right? So, which are the major chemical compound which gives the efficacy? You mentioned phenolic compound. So, what are the names which may be responsible, or maybe other than phenolic? So, the names, if you can reveal, it will be better. Yeah.
Thank you very much for the question, Mr. GP. No, actually, I do some LCMS analysis too, but I don't put it here. So it's actually flavonoid, from flavonoid. I do some HPLC too, so flavonoid is lutein, something like that.
Okay.
Yeah, the flavonoid actually, and phenolic too. Yeah.
Okay. Thank you.
Okay, you're welcome.
Thank you, Dr. Patel. Is there any question, please? Maybe I have one question for Rizka Bella before we proceed or accept other questions from the floor. You did the study for acute oral toxicity study of kebab grass using ethanolic extract, right? So, and the result you saw that even in the highest dosage, it's 5,000 milligram per kilogram, is still safe, right? Do you think you need to like give higher dosage or just enough to make sure it's safe, or there is no toxicity?
Can I use in bahasa? Dalam peraturan dalam BPOM untuk toksisitas akut oral itu tertinggi di 5.000 digunakannya. Jika lebih dari 5.000 itu tidak perlu dilakukan karena sudah termasuk aman. Tapi perlu uji lanjutan seperti subkronik dan hispatologi dalam toksisitas. Thank you.
I try to translate in English, if I'm not mistaken. So it's not necessary to put the higher dosage since it's 5,000 is already safe, right? But we need to do another analysis, check in the subchronic dosage and other type of toxicity dosage, right? Okay. Thank you. Is there any questions? If there is no more, I think it's time to wrap up this session. Thank you so much again for Professor Endarti, for Rizka Bella, and for Bunga Anggraini. Thank you so much for sharing the research and studies. One more time, we give a round of applause and have a wonderful day. Thank you.
Thank you so much for the insightful topics, and we respectfully request all participants to remain in the room, and we will wait for the other participants to return. As an announcement for the best oral presentation and best poster award will come shortly.
Di tengah perubahan zaman yang begitu cepat, Universitas Nasional tetap teguh berdiri. Sebagai universitas perjuangan, UNAS hadir mengemban komitmen mencetak generasi unggul, berintegritas, dan siap bersaing di tingkat global. Lebih dari 7 dekade perjalanan, UNAS terus meneguhkan jati diri sebagai pelopor pendidikan tinggi Indonesia. Tempat di mana ilmu dan nilai tumbuh bersama semangat kemajuan bangsa. Didirikan pada 15 Oktober 1949, UNAS menjadi perguruan tinggi swasta pertama di Jakarta dan tertua kedua di Indonesia. Lahir di masa perjuangan, universitas ini didirikan oleh para intelektual dan pejuang kemerdekaan. Termasuk ayah dari Presiden Prabowo Subianto yang percaya bahwa pendidikan adalah jalan menuju kemajuan bangsa. Dari semangat itu, UNAS tumbuh menjadi institusi pendidikan yang kokoh. Melalui ribuan alumni yang kini berkiprah di berbagai bidang, pemerintahan, akademik, hingga dunia profesional, UNAS telah memberikan kontribusi nyata bagi bangsa dan dunia.
Universitas Nasional berkomitmen menjadi universitas perjuangan yang berdaya saing internasional. Kami terus berinovasi dalam pendidikan, penelitian, dan pengabdian kepada masyarakat, serta memperluas jejaring global untuk menghadirkan pengalaman belajar yang relevan dengan masa depan.
Kini, Universitas Nasional berkomitmen menjadi universitas yang berdaya saing internasional. Kami terus berinovasi dalam pendidikan, penelitian, dan pengabdian kepada masyarakat, memperluas jejaring global untuk menghadirkan pengalaman belajar yang relevan dengan masa depan. UNAS memiliki 8 fakultas wadah pengembangan ilmu dan inovasi bangsa. Fakultas Ilmu Sosial dan Ilmu Politik, Fakultas Hukum, Fakultas Ekonomi dan Bisnis, Fakultas Bahasa dan Sastra, Fakultas Teknik dan Sains, Fakultas Teknologi Komunikasi dan Informatika, Fakultas Ilmu Kesehatan, dan Fakultas Biologi dan Pertanian. Setiap fakultas dikembangkan sesuai kebutuhan industri dan perkembangan global, didukung oleh tenaga pengajar profesional dan fasilitas pembelajaran yang modern. Universitas Nasional menerapkan sistem pembelajaran berbasis outcome-based education, memastikan mahasiswa tak hanya memahami teori, tetapi mampu mengubah pengetahuan menjadi karya nyata. Untuk mendukung itu, UNAS menyediakan laboratorium riset, studio kreatif, ruang diskusi interaktif, dan lingkungan belajar yang mendorong kolaborasi lintas disiplin. Kami memperluas kerjasama akademik dengan berbagai institusi nasional dan internasional agar mahasiswa memiliki akses pada pengalaman belajar global yang membentuk karakter profesional dan berwawasan dunia. UNAS memiliki 3 kampus utama yang saling terhubung. Kampus Pusat Pejaten, Pusat Kegiatan Akademik dan Administrasi, Kampus Bambu Kuning, Pusat Riset dan Pengabdian Masyarakat, Kampus Pasca Sarjana Ragunan, ruang tumbuhnya riset dan pemikiran ilmiah tingkat lanjut. Ketiganya menjadi ekosistem pembelajaran yang menumbuhkan kolaborasi, kreativitas, dan intelektualitas. Sebagai universitas modern, UNAS menjunjung tinggi nilai pionir, fondasi dari setiap langkah pengembangannya. Perseverance, berjuang menyelesaikan setiap tugas dengan sepenuh hati. Integrity, jujur, beretika, dan dapat dipercaya. Optimistic, melihat tantangan sebagai peluang untuk berprestasi. Networks, membangun koneksi global untuk kolaborasi berkelanjutan. Innovation, melahirkan gagasan kreatif yang solutif dan berdampak. Respect and collaboration, menghargai perbedaan dan bersatu untuk tujuan bersama. Nilai-nilai inilah yang menjadi roh Universitas Nasional, membimbing setiap langkah untuk terus berkarya, berkontribusi, dan membawa nama bangsa ke kancah internasional. Dari masa perjuangan hingga era digital, semangat UNAS tetap sama. Mengabdi untuk ilmu, berjuang untuk bangsa, dan melangkah menuju dunia. Universitas Nasional, Universitas Perjuangan, Universitas Masa Depan.
Hari ini kami berkumpul di sini dalam rangka restorasi ekosistem mangrove. Kami mohon kepadamu ya Allah, keridhoan-Mu, limpahkanlah ridho dan berkah-Mu dalam acara kami ini ya Allah.
Mudah-mudahan acara ini bisa dicontoh. Ini satu acara yang saya kira diinisiasi oleh Bapak Duta Besar Malaysia. Mudah-mudahan Duta Besar menyambut dan meneruskan sehingga banyak pohon-pohon mangrove yang kita tanam di seluruh penjuru Indonesia. Mudah-mudahan didengar oleh teman-teman Duta Besar.
Semoga inisiatif sebegini menjadi nilai tambah pada kerjasama kedua negara dalam mencapai tujuan pembangunan berkelanjutan atau Sustainable Development Goals 2030. Semoga ia juga mengeratkan lagi tali persaudaraan di antara Malaysia dan Indonesia.
Satu program yang sangat bagus kerana ianya bertujuan untuk memelihara alam sekitar supaya menghindari dari terhakisnya pesisir pantai, dan sudah tentu ia akan memberi kesan yang positif kepada masyarakat setempat juga, dan sudah tentunya lingkungan hidup itu sendiri. Dan kita sangat berterima kasih dapat kerjasama dengan UNAS dalam program pada kali ini. InsyaAllah kita akan dalam program ini 10,000 pohon akan ditanam dalam masa 6 bulan.
Fakultas Biologi dan Pertanian Universitas Nasional mengadakan seminar dalam rangka Hari Keanekaragaman Hayati Internasional. Acara ini dilaksanakan bersama dengan Yayasan Kehati yang sudah menjalin kerjasama serta penandatanganan MOU dengan Universitas Nasional, yang dihadiri oleh siswa dan guru di beberapa SMA di daerah Jakarta Selatan. Dengan judul Keanekaragaman Hayati untuk Kesehatan Mental dan Pembangunan Berkelanjutan, seminar ini membahas betapa pentingnya kesehatan mental dari peran keanekaragaman hayati. Acara ini dihadiri oleh yang pertama yaitu Dekan Fakultas Biologi dan Pertanian Dr. Fahrudin Mangunjaya MSI, Guru Besar Universitas Nasional Profesor Dedi Darnaidi, dan CEO Seir ID SSGN Fellow CTS SIPB Dr. Sunarto MSc PhD, yang akan memberikan materi pada acara seminar ini. Setiap pemateri memiliki topik yang berbeda untuk dipresentasikan, yaitu yang pertama ada Profesor Dedi Darnaidi selaku Guru Besar Universitas Nasional dengan topik membahas tentang evolusi Indonesia selama 50 juta tahun. Mulai dari awalnya terbentuk pulau-pulau di Indonesia, flora dan fauna, budaya yang kental dari kebiasaan, biooperasi proteksionisme dan nasionalisme,
tanggal 28 November 2024, Fakultas Biologi dan Pertanian melepas mahasiswa Baik Seperti Warrior bersama Patu bekerja sama dalam upaya sosialisasi dan penanaman pohon dan matahari pohon dan mengajak beberapa siswa SMA. Saya berharap bahwa kegiatan ini menjadi satu momentum di mana para siswa selain dapat belajar dan memiliki hikmah bagaimana belajar di alam bersama mahasiswa Fakultas Biologi, juga bisa mendapatkan pengalaman bagaimana meneliti dan mengenal satwa, flora atau fauna di kawasan-kawasan lindung yang ada di Jawa Barat. Terima kasih. Saya harapkan ini akan menjadi semangat buat kita semua dan menjadi suatu pengajian bagaimana kita tidak berkata-kata. Assalamualaikum warahmatullahi wabarakatuh.
Dari program kehutanan Yayasan Kehati. Hari ini seru dan berbahagia banget bersama teman-teman Biodiversity Warriors Universitas Nasional di Ciwidey, Kabupaten Bandung bersama Yayasan PGR dan Yayasan Kehati pastinya mendukung teman-teman BW UNAS ini ikut menanam tidak hanya menanam, pastinya memelihara sampai pohon-pohon yang ditanam ini itu bisa tumbuh besar, menjadi tajuk-tajuk yang sangat baik dan menjadi hutan kembali.
Kita berusaha untuk salah satunya ya kita menanam pohon yang pertama adalah untuk memberikan dampak baik bagi lingkungan. Gitu. Lalu kedua ini sebagai legacy ataupun masa depan bagi teman-teman generasi muda. Nah, BWKP mensupport
Welcome back to the third International Conference on Natural Products and Chronic Diseases 2025, Natural Products for Quality of Life, Advancing Global Health and Sustainability. Ladies and gentlemen, we will now have a short free time. Please. Check, check. Ladies and gentlemen, we will now have a short free time, and please feel free to move around and explore the poster displayed on the stage area of the room.
based on ini aja bilangin based on giving question. Hah? Ya udah kok berapa? Ya terserah. Asking question ya. Asking question. Hah? Korea. Oh udah. Oh Indonesia dua-duanya. Jangan dong. Satu aja. Nanas. Nggak boleh. We have no break. The very special highlight of the speakers. Oh nggak boleh. Of dress. Ya udah.
Ladies and gentlemen, we have now reached a very special highlight of today's conference, the announcement of the Best Oral Presentation Award and Door Prize. This award is presented to acknowledge outstanding research and meaningful contribution to the academic community. After a thorough assessment, we are pleased to officially announce that the Best Oral Presentation Award goes to M.A. Rachel V. Parken, Dr. Jelita Patel, Dr. Shreya, Indah Trilestari, Dr. Jitendra Patel, Dr. Shreya, Dr. Indah Trilestari, Elvira Nura Effendi, Akram Abdul Wahab, for joining us by virtual channel, Repo Raham SPD MSI, please come up to the stage. And we can invite to Professor Dr. Ernawati Sinaga MS APT to give the reward speech, please. Moving the next agenda, Best Poster goes to Dr. Hiji Sim. Rizka Aprilia Aldini Esraf. Sanjara Sindhukono. come to the end of this event, the third International Conference on Natural Products and Chronic Diseases 2025, Natural Products and for Quality of Life, Advancing Global Health and Sustainability by Universitas Nasional. On behalf of the organizing committee, we would like to express our deepest gratitude to all distinguished speakers, moderators, participants, and everyone who has contributed to the success of this event. We truly appreciate your active presentation and enthusiasm through this season. We hope today's conference has been insightful and will inspire future collaboration and innovation. Once again, thank you for being part of this event. Have a wonderful day and see you again at our next international conference. Wassalamualaikum warahmatullahi wabarakatuh. We remind to all participants